catalyze excellency in higher education institutions across the country, including all IITs, IIMs, Central Universities, IISc Bangalore, IISERs, NITs, IIITs, progressively extended to reputed state universities. It is a dynamic program that promotes participation of international faculty as a visiting professor or professor of practice, offering specialized 5 or 10 days courses to enhance academic collaboration and knowledge exchange. The first GIAN course at Panjab University was conducted in 2016. Till date, 28 courses have been successfully organized, and we look forward to conducting many more in the future. Panjab University has submitted 40 new proposals under Phase 5 for the upcoming financial year 26 to 27. I wish you all an enriching and insightful learning experience with Dr. Paj. Thank you.
Thank you, sir. May I request Professor Amit Rawat to propose a formal vote of thanks?
I would first like to thank the foreign faculty who graciously agreed to come for 5 days to India for teaching us more about bioinformatics and large-scale data analysis. So the program was initially designed only for genomics, but then we realized that transcriptomics was also very important. So we'll also be doing some exercises on bulk transcriptomics and single cell. And for that, I thank Dr. Lopez and Dr. Bonnie Day for taking all the trouble. And as Bonnie has been telling me that Frank has been working for the past 1 month religiously to put up a good course. So I hope you benefit from that. And you also put in effort to learn as much as you can in these 5 days from him. I would like to thank Professor Gurjaspreet, the local coordinator for GIAN. He has been very encouraging and helping us whenever there is a call for proposals. He reminds us, and when we get stuck somewhere, he helps us with that. So thank you, Professor Gurjaspreet, for that. I would also like to thank my course coordinator, Manpreet. She has been doing a lot of behind the scenes, arranging, booking hotels and doing all the other logistics, working out the logistics for this workshop. And also my other colleagues, both in the lab and the clinic, who have been helping out with the workshop. And finally, and not to say the least, the participants. So we had got an overwhelming response for this. And I'm sorry if we could not accommodate all, because it's a hands-on workshop and we have got limitation because we have to have computational space, so we limited it to 40. So most of you are from Chandigarh, but I believe that few of you have also traveled all the way from something Patna and other parts. So thank you for making it for this workshop, and I hope it will be a great learning and enriching experience for you. Thank you. Thank you, everyone.
हाँ जी सर।
एक कभी जब आपको मेल आएगी 5 मिनट में 10 o'clock पहले आई हुई थी 10 o'clock तो 40 पार्टिसिपेंट्स और लिंक टू भेजें एंड वो लिखेंगे यस स्टार्ट हो गया यस ओके ठीक है सेकंड सभी विल केयर बी रखेंगे ठीक है और उनकी फोटो कॉपी या मुझे व्हाट्सएप करते रहेंगे ठीक है बाकी मिलते रहेंगे मैं ऑनलाइन रहूँगा ठीक है थैंक यू
I think it's the distance between.
Yeah, I think you have to keep it here.
Yeah, we keep it some distance now.
Yeah.
I think it will be fine.
Let me just do a mic check. Hello, can you hear me now? Is this okay? Or is this okay? No? I'm doing a mic check. Hello, hello. I think, can we use the other mic? Darshan? Darshan? Oh. तो wired है, तो नहीं होगा. ठीक नहीं आ रही है. अब आ रही? When I talk like this? No, but here also नहीं. Now not ना. ये ठीक नहीं आ रहा, बहुत ज़्यादा फटफट फटफट
मुंह के सामने बात करना, कॉलर माइक है।
नहीं बट स्टिल इट्स नॉट देखो इन दिस
हेलो 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 हेलो।
स्पीक लाइक दिस
ओके, स्टिल अभी के लिए
नो बिकॉज व्हेन आई वाज टॉकिंग ना
हेलो
मुझे बहुत
हेलो।
फीडबैक आ रहा था इज इट वेरी इनकन्वीनियंट
I don't know. Hello, hello. The popping, yeah. Hello. This time is not going to work. I have to have the laptop there, so it's going to be a little bit complicated to.
Why? What happened? हाँ हाँ, ये कन्वीनियंट नहीं है, फॉर सम रीजन वर्क नहीं कर रहा है। हाँ हेलो, सुनाई दे रही है वहाँ बैक में सुनाई दे। सो व्हाट्स द व्हाई यू कांट डू इट एज दिस देयर, व्हाई यू कांट डू इट?
Cable is too short.
you can take this here
Okay, then you have to, yeah, that's fine.
See, when we're coding, maybe we can have to see.
Yeah. So let's start. Okay. So let's start with a little bit self-advertisement. So as I mentioned, it's me, Bonidi Kumar, and then this is Francis Lopez. We are going to, help you to learn a little bit about bioinformatics for next 5 days. So we have at least 4 faculties today for next 4 days to deal with this course. Dr. Amit Rawat is here, Manpreet is here, and me, myself, and Francis. We will be also joined by Jomin Choi. He will be here on Thursday, hopefully, and he will be also leading variant analysis, that part of the course along with Francis, and he will be here on Thursday. So he's from Department of Biomedical Sciences from Korea. He used to be in Yale previously, but now he is a independent faculty in Korea. So he will be traveling all the way from Korea to join us on Thursday, and he will be there with us on Thursday and Friday. Apart from that, you will have few interesting talks to attend. Every day 5 to 6 we have a talk, which basically to give you a sense that how you can use NGS for a real life case. So we will be, so first we have Ganeshwar. Hopefully today I am just still waiting for his response. If I do not hear from him, and unfortunately I did not realize that today is Chhath. So those people who are from Bihar and UP, and he is from Banaras, so from Gorakhpur. So essentially he might be within that Chhath thing, and I could not hear from him yet. So if that is the case, then probably I will be talking, if time allows, then I will be talking about pediatrics, using NGS for pediatrics and newborn screening, but otherwise Ganeshwar will be here to give a talk. Our director for bioinformatics at Yale Center for Genome Analysis, James Knight. Jim will be talking more about best practices in bioinformatics and some of those aspects of data analysis. Then we have a clinician investigator, Rick Pierce, who will be talking about MODS and how the actually multi-organ dysfunction in neonatal ICU kids, and how he is using genomics to study those kids, and especially the origin of MODS in those kids. Then we have Madhav Menon. He was an alumni of this place, PGI Chandigarh. He did his MD and residency from PGI Chandigarh. Currently, he is also director for kidney transplant center in Yale, and he will be talking about how he is using NGS based analysis for mismatched loci to identify whether transplant rejection is happening, and you can see some of those analysis where you can figure out if those rejections at very early stages, even the stage when you cannot detect many antibody related to some of those current practices that we are doing. So he'll be talking more about that, and of course Jomin Choi, he himself will be giving a talk on how he is using NGS to study rare diseases. And so you will have a series of talks you will be hearing where you will how the NGS is being used. Then this course is actually, there are more finer points to this schedule, but this is a kind of a broad outline. We will be introducing today you to with some basic bioinformatics tool, and then from there we will be going to experimental design and transcriptome analysis tomorrow. We will consolidate on that theme on Wednesday with bulk RNA-seq. So you will see some of the basic thing on Tuesday, and then we will consolidate that idea more into bulk RNA-seq on Wednesday, and Thursday we will be furthermore going one more step further, and we are actually going to learn single cell RNA sequencing, and finally on Friday we will be doing variant calling and interpretation of structural variants, single cell SNPs and all. So as you see, this workshop is actually structured in a way, it's a progressively increasing difficulty format. So, and then you need to be here every day to learn if you want to make use of it completely. So who we are? So me and Francis, both of us are from Yale University, and if you don't know Yale, Yale is actually in top 10 universities in the world and second ranking university in US. And so I know ranking doesn't mean anything, but still just to mention that, I say to put things in perspective, that's why I just mentioned that. So these universities, Yale University along with Harvard, Princeton, Brown, they all comes under a group of universities called as Ivy League. Ivy League, you may think it's because people are envious about it, but no, that has nothing to do with that. It's just because that was a sports league which they were part of, but they are the oldest school in US, and they are one of the best schools that US offers as a part of higher education. This is the second oldest university in United States. It's almost 250 years. Second or third? Which was the second then? Harvard then?
Harvard is the second.
What was the first?
It's a college called John Mary something.
Okay. So I'm correcting. So it's a third. So they started. So the history is they started Harvard, for imparting Christian education when Britishers came to US were colonized. And at some point they realized that Harvard is becoming way more liberal than they wanted it to be. So they established Yale to again emphasize on the Christian education. But unfortunately, Yale became more liberal than Harvard, and it is one of the most liberal, liberal-leaning universities in the world at this point. Okay, there is a connection to India with Yale University, and it's not a good connection in a way. So when the initial building of Yale was being conceived, and they were looking for funding, this funding came from a person who was part of East India Company and posted in Madras. So he took bales of cottons from there and sold it in European market, and that money was actually the basis of the first building of Yale. Second thing, and then he actually collected a lot of riches from exploiting the local people, and that was also actually became part of Yale's some of the initial funding. And so there is recently there was Yale actually instituted a project where they were studying about slavery and how the slavery, how actually Yale and slavery was actually interacting to each other. And one of the things they apologized to, after the project was completed, they published a book about it, and they apologized to Indians and as well as some of the other people who were part of those oppressive part of. But it was not like Yale did something, but it was some of the money that came from such a background, which at that time it was not. So this is the thing. And now also Yale offers the advanced training for Indian administrative service professionals. So those who pass IAS and complete everything, and after 5-6 years of their work, like their job, they go for advanced training, and one of those trainings are conducted in Yale still now. So they almost, it's I think 8 weeks training that happens in Yale. So it's so Yale is that way. It's interesting. So we are located in a place called as New Haven. New Haven is almost 1.5 hours from New York and 2.5 hours from Boston. So we are between New York and Boston. So those are the references probably you are familiar with. So it's an old town. It's our okay. So Yale has a big endowment, like it's a private university, so a lot of money we get. We have current endowment level is approximately $42 billion that we Yale has. And then we have around 6,800 undergrads. There are 8,750 postgrads and around 6,000 academic staff. So, and Yale is actually divided into 13 schools, including School of Medicine is one of them. And Yale's law school is one of the considered as one of the top in the world even. So there are 2 law schools which are competing for this honor. One is Harvard and second is Yale. The difference is Harvard people go for corporate laws, while Yale people are more idealistic and they go for Supreme Court clerkship and those kind of things, and pro bono, non-profit. So a lot of idealism is there in the Yale law school. One of the interesting thing about Yale is that Yale gave almost 7 presidents to United States, and this administration, interestingly, the vice president and the second lady, which is vice president's wife, Usha Vance, both of them are Yale alumni, and they did their law school from Yale. So, and you know that Usha is from India. So again, that is something. And then famous Indiana Jones movie, Kingdom of Crystal Skull. This was shot in Yale. So those who are fan of Indiana Jones, genre of movie. So it was third movie, the Indiana Jones.
Third or fourth.
Yeah, one of those. So that was actually shot in Yale. So you will see that. Now coming to the Yale Center for Genome Analysis, which I am the director for operations, as well as Francis is there as a bioinformatics associate director. We are located in West Campus. So Yale has a main campus and also a West Campus, which is almost 8 miles. 8 miles means 13 kilometers away from the main campus. We have shuttle actually frequenting between these 2 campuses. Every 15 minutes we have shuttle to go from one place this to other. So we have a dedicated around 7,000 square feet of sequencing facility, and as we said, that this is a West Campus. This is how the West Campus, Yale West Campus looks like from the aerial view. We are located in, this is just the geographical location, we are located in New Haven, and this is New York actually, Long Island, and we are here. So this is just to orient you with the geography where we are with. So as we said, like we have 7,000 laboratory space, and actually we have another 4,000 square feet area which is for office and other space. So we have total 11,000 square feet of area. We have 35 people working with 8 PhDs and a lot of masters and bachelor's people there. Our data, whatever we are producing, that is live streamed to a high performance computing system, and this computing system consists of around 400 nodes and 4,500 or 4,500 cores, and each of those cores have 32 gig RAM and 16 GPUs are also there. So, but we are not using much of those GPUs for our work yet. But and then we also have 2 Dragon servers which is GPU accelerated, and we are yet to use that. Again, the older version of our HPC was cooled by like the what they call door cooled. They were door cooled ones. So we were spending a lot of money for electricity bill. Almost our per month electricity bill was $100,000, slightly over $100,000 just to pay for electricity for cooling the facility. So for the new HPC when we bought, we moved to a water cooled processors, which means like the water will flow through the processor and it will take out the heat. So that means we reduce some of the cost of our electricity bill through that. Now we are moving to a larger center, which a collaborative effort between Yale, Harvard, Tufts, Boston University, I think MIT, and one other. So it's a collaborative center of data center we are creating, which is called Mass Green, and it's in Massachusetts, and we are moving all our infrastructure to there. But the key point is that we have a 10 gig optic fiber which transmit data from our sequencing center to directly on a real time basis to the data centers, so that data is stored and processed there. We offer a variety of services from our facility, and this is kind of a laundry list of that, and you can see anything that you can think of we are doing. Apart from this, we are also doing a lot of clinical services. We are doing rapid whole genome sequencing. Rapid whole genome sequencing, if you are not aware, it is for NICU, PICU kids, those who are severely ill NICU kids. We sequence data and report our analysis within 30 hours. So that means from the blood collection to data generation and reporting, it is done by 30 hours, within 30 hours, and that report goes back to clinician for clinical interpretation or clinical action, not interpretation, clinical action. We have a pretty good solve rate. We are almost at 50% solve rate, which we are really proud of. Probably those kids are really sick, so we usually get a reason why they are sick. So, and this is one of the fulfilling part that we do those kind of operations. So other than that, we also do tumor profiling. So hematological tumor profiling we do from our center. We also do profiling for neurological tumors like glioblastomas and meningiomas and all. And then also we are now currently working on solid tumor profiling with the FFP sections. We are just waiting for the final regulatory approval, but we have done most of the groundwork for us. So once it is done, we'll do that. Other than that, we do analysis for newborn screening and newborn kids with dry blood spot. So a lot of these things what you are seeing with the dry blood spot based is for the newborn screening programs. So we support some of those newborn screening programs from YCGA, and we use all this exome and whole genome sequencing from blood. So I don't know much about the regulations in India, but in US you cannot take, like, unless there's a reason to draw blood from a newborn baby, you only can take like a prick, a heel prick, and you can take only 2 prick cards. So that is the only you are allowed with. So that means you are working with a scarce material and you have not much to do. So we have done a lot of technological innovations on that side, from the extraction of DNA to library prep and sequencing and everything. So last 2-3 years we have done a lot of those things, and then we are now at a stage when we can do these things. Okay. So history. So it all started as a KEC. We got a grant in 2008, and Dr. Shrikant Mane and Dr. Rick Lipton. Rick is currently the president for the Rockefeller Foundation. He was the person who first discovered hypertension gene, a very accomplished scientist. He was chair of genetics department at Yale and currently president of Rockefeller. So they both started with the NIH grant in 2008. And so we started YCGA, and AIL actually provided us with a $10 million grant for establishing this. So which we, and still we have that money because we spend that money on instruments and then we recover the cost through our services. So we still have most part of that money that AIL gave us. Then the first NGS paper on clinical diagnosis came in 2009. This was the science paper, and this was the beginning of precision medicine. So first report of a precision medicine where they have correlated a gene and with a clinical diagnosis was this paper, and this came from our lab. And we were also instrumental in developing exome sequencing. So a lot of those, and Francis was one of those earliest people who analyzed those data, I believe. Is that right? Yeah. So these were the people who initially start. So we are doing microarray, and then they used that solid platform and took into a liquid phase, and they hybridization based analysis. They actually established those protocol, and Francis and his group were at that time doing the basic analysis. So they established exome sequencing. So you can see some of those early remnants of historical people who were involved in developing exome sequencing. So and we were proud to be part of it. So 2010 we bought our first Illumina machine, which was a GA2X machine, and it became operational, and we established a CLIA. CLIA is basically a certification that you need to, if you are running a clinical sample and you want to report it. So if you want to do and this is given by a College of American Pathologists, so which is CAP stands for that, and this certification you have to maintain. Every alternate year you have to undergo a certification process, and every alternate year you have to also go through state health department recertification. So this is a very extensive, laborious administrative process, and unfortunately I have to do that every year after year. And you can see like this much area, like almost these 2 rows of files are there. Still they want papers, like you have to print it and keep it. They won't allow, they don't want any and in fact actually last COVID time when they were doing this CAP inspection, they told us that okay, you can submit some of those things online, and that was more complicated than me giving it to the paper one. So and we have to maintain all those, like since 2010 all these papers are there and you have to maintain it. So it's a process. So we got our certification, and then and we have to keep validating if we are doing any new assay for patient care. Okay, and then our first bioinformatics group was started in 2012, and we got our first Illumina sequencing, HiSeq 2005, a second Illumina machine, which was the latest at that time was 2500, and then 2015 we got our next Illumina machine 4000, that was a new one, and then 2017 we got 6000 Illumina 6000, and 2017 itself we got our sequencing a 10x machine for single cell RNA sequencing, and 2022 was another year when we just came out of our COVID, and then we established spatial technologies, and I don't know how many of you are aware of this. So this is a kind of a technology where you analyze single cell on a tissue context. So you will be also looking at the position of cell, and then you have a single cell transcript analysis. So we have 4 different spatial platforms which we established in 2022, and in 2023 we purchased our most latest state of art NovoSeq X Plus and a NextSeq 2000 at that point. And so we have, so this is kind of our history, how we published more than 1500 papers, and then many of them are in Nature Cell Science, and we attracted almost more than $500 million in grants and funding. So, and we actively write grants. We also support other people to write grants for genomic space. The one of the thing that I don't know whether you could perceive it from the presentation, from the history, that every time a new technology is introduced, YCGA is at the forefront of acquiring it and providing that to the researchers. Because unless you give the state of art technology, you cannot leverage the cost and other things, that advantage that comes with that advancement. So from the 12 GA2X sequencer, which was the beginning of YCGA, now we have 2 NovoSeq X Plus. So NovoSeq X Plus is a state of art Illumina sequencer at this point. We almost generate around 7.5 terabases of, 15 terabases of data in a 48 hours run, which is actually, if I want to convert this into billion bases, we generate 52 billion reads every 2 days from YCGA, and we are generating around 1 petabases of data every year from 1.1 petabases of data from YCGA. Approximately, we are running 30 to 36 flow cells a month, which is like around, say, somewhere between 6 to 8 flow cells a week. So this is the scale of operations that we are doing, and because of that, we are able to achieve scale of economy. And our current sequencing cost is $1.05 for 1 million reads. So I need to put that also in perspective. One burger, so don't consider McDonald's of India. The McDonald's of US is the cheapest food that you get in US. So the cheapest low income people's food, you can say, is actually cost $6 for the cheapest burger in McDonald's at this point. And so you are getting a million reads for 5 times cheaper than a McDonald's burger. So this is the kind of scale of economy that you can achieve through having the new technology and the throughput that we can think of. So this also created a situation where people started moving away from exome sequencing, and now they are doing whole genome sequencing, because now the cost of exome sequencing and whole genome sequencing are almost comparable, like a few $100 more than, which means like you have more data, and then you are, so we are seeing that conversion, and we are also currently working on whole genome sequencing for clinical applications, and we are doing some pilot studies on that. So as I mentioned, all these technological innovations led to a decrease in cost, and we are actually capturing this decrease in cost by introducing new technologies and new sequencing platforms at YCGA. So YCGA has multiple sequencing platforms at this point. We have one MySeq, we have one NextSeq, we have 2 NovoSeqs, we have one Element AVITI. By the way, actually I wanted to mention, Dr. Rawat, that this is a platform that probably you should look into, okay, instead of your NextSeq 2000. Element AVITI, the cost of sequencing is almost equal to the 6000, NovoSeq 6000. So you will get a medium throughput sequencer at the sequencing cost of a high throughput sequencer. So this is a very good platform to do. Then Nanopore MinION, we have a PacBio Ravio. Ravio is the latest PacBio machine, and we also have a PacBio Sequel 2. Yeah. I was mentioning about spatial technology. We have at least 5 different technologies, and we will be seeing some of this when I'm going to teach you more about the next-gen sequencing technologies in 2 days, different days. One I think today, and then other is on Thursday, I believe. Okay, so we have Visium, we have Curio, we have Atlexomics, we have Cosmex, and we have Xenium. So we have 5 different platforms for spatial technologies at YCGA. And with that, I would like Francisco to talk more about the workshop.
Sure. Well, you already heard about the workshop a little bit while we were waiting, right? So what is the workshop, right? We want to be a hands-on workshop. So we want you guys to be able to do some analysis or use the tools, etc., right? But you also notice, I noticed in the audience there were a few people mentioning single-cell RNA-seq, other people even mentioned epigenomics or things like that, right? So the field is so vast that we are not able to cover everything, right? We are not going to be able to cover every single analysis. Even if we talk about single-cell RNA-seq, we could have a single workshop about it, right? So this is not about it. So we are not able, we are not going to be able to see everything, right? One of the ideas that we have about, okay, what topics we're going to choose for the workshop was about, okay, what are the things we do the most at YCGA? So the things we're going to see, like bulk RNA-seq, single cell, and variant calling applications are probably the 3 most applications we do at YCGA, right? I would say, I don't know, Bonnie probably know better than me, but I would say at least 80% of those falls into those 3 categories, right? Single cell is, of course, is now the new fashion, and probably we do a lot of it. But bulk RNA-seq, we still do a lot of it, right? Maybe not at the cost level, because it's really cheap to do bulk RNA-seq compared with single cell, but at the number of samples, I wouldn't be surprised that it's still quite common. And we still do a lot of whole exome sequencing and whole genome sequencing. So that's the reason we pick those 3 topics. So, and this is also really important, right? The idea of if we want to be a useful workshop, right, we need to pick things that not only about the technologies or the analysis types, we want to teach you something that you can take from these and use it in something else, right? So we talk about epigenomics, I think somebody mentioned it, right? So if you can, there are a lot of commonalities in all the analysis we do that you can take them and use it somewhere else. It could be using R. You can use R not only for single cell, you can use it for bulk and other type of analysis, or even just producing plots for your papers. Or if we talk about Jupyter notebooks, you're going to be able to use them for anything you do, right? Probably in the lab regarding analysis. And within the analysis types, right? So aligning sequences to a genome is something you do for a lot of things, not only for bulk or variant calling. You're going to do it for single cell or even methylation analysis or anything you do. There's a lot of commonalities in all the types of analysis. We give you a little survey before the workshop, right? So this is where the answers about the knowledge of R. So this is a big challenge, right? So the people that comes to workshops, and this is common, right? We see this all the time, that when we organize a workshop, you're going to have all kind of levels of knowledge, prior knowledge before coming. So, and it's complicated to prepare a workshop where you're going to, make everybody happy. So if you do it too complicated, then the people that's new to the field, they're not going to learn much. If you do it too easy, then these people get bored. So one thing that I recommend is we're going to do exercises in groups, right? So, and individually, both. But, don't hesitate to help somebody. So if you fall in the side of, oh, I already know how to do things in art, and you see somebody struggling, please help me. So I think in that way, so if you already know the topic, you can help somebody else and maybe learn something else too. If we talk about, so the goals of the workshop, right? So one of the things, you're going to hear talks, right? With those talks, we will offer, so the type of resources that you have to do with the NGS, right? It's not only about sequencing, there's all applications of that NGS. So the talks are going to give you some ideas about the different applications we use NGS for. One thing that I noticed, I think it's important, and maybe it's boring for everybody, but I think it's important to understand the data formats that we use when we do data analysis, right? I know this is not interesting. Sometimes the only thing you want is to run a pipeline, get your results, and that's it. But there's a lot of troubleshooting when we work in data analysis, a lot. And part of it, you need to understand the type of data formats that we use. What we said before, right? We're going to try to give you tools that you can make use not only for the things that I mentioned, the data types that we're going to be working, so you can extrapolate to anything you do about data analysis. Well, of course, that's the main idea. A lot of people come here, right? Half of the people come with the idea, I'm going to be able to do my own analysis. Well, I don't know how much you're going to learn here, but I hope you learn quite a bit, at least to get your feet wet on the field and learn more things in your own. And this is something that I always mention. So I'm part of the offering service most of the time, right? We help people to do data analysis, or we collaborate with people, with investigators that they only work in the lab. And there's always a barrier of communication. So one thing that I always say is invite people that only work in the lab is to learn this, right? How to communicate with a bioinformatician. If you know the language, if you know how to communicate, so both sides of the equation, they're going to learn a lot of it, right? They're going to be able to produce better data and better analysis. We're going to talk the first tool we're going to be using. It's not related to data analysis, but this is a website. Are you familiar with Poll Everywhere? Do you use it in classes? Oh, you use it sometimes? Okay, so if you don't mind, go to this website. So, and if you can, you should bookmark it, because we're going to be using it through the talks maybe, and at some point we're going to be using this website. Oh, you can use even your cell phone, by the way, for this. You don't have to use a laptop, but if you go to the website, keep that tab open if you can, in your phone or your laptop. I don't think it needs to be uppercase. I did it uppercase so you can see there's always confusion about the L, if it's an I or not, if it's lowercase. Yeah, everybody is in the website. So why we use this? So oh no, there's no password for this. There's not going to be password. Don't worry about it. No. Oh, for the wireless. Oh, okay. So it's immunology 5G, and the password you know better than me. I forgot.
So the immunology 5G and the password is LOGY, all small case. LOGY.
Oh well, I shouldn't put it. Oh, I don't know. Yeah, you can. So
Can you open it?
So if you want to type it, you want to type it here.
Yeah. So both of them will work. There is one called immunology 5G and the other one is 4GIS, 2.4. So you can connect either. If you are able to connect 5G, it's better. The password is same.
So everybody has wireless. You can say no if you don't. Good. Should we move on? Did you access the website? It's going to ask you for a username. I think you can put whatever you want or ignore it. Any questions we ask are going to be completely anonymous. So can we test this? So we're going to ask questions like this during the workshop. But I was asking Bonnie what controversial question could I ask, and say, talk about cricket, and say, I don't know. I don't know who these people are, so you can say whatever. I'm going to believe whatever your answer give me. So try to answer the question. It's not updating?
It's not updating.
No? Huh. Oh, I see it. The funny thing is I see it in my computer, but not here. Okay, let me try to go back. Why it's not? Well, I can tell you, Sachin Tendulkar is winning with 36%. Is he the best? Is it
not showing there?
I don't know. I'll check it, but it's not. It's showing in my computer, but not on the screen. That's a funny thing. Probably if I do this, let's see.
Maybe you can refresh it.
I'm trying, but I don't know why it's not.
Are they able to use it now?
Why it's not? Huh.
Why don't you refresh it and see?
We'll figure it out. I'm going to ask you one more question, if you don't mind. Is if you perform, we sent an email about pre-workshop materials. So how many people did perform the pre-workshop materials? What? No. Oh, I'll see it on the screen.
Yeah, you can actually register there in your code. You can see that on your screen.
I can see it in my screen.
Why it is not showing?
I don't know. We'll do a test later. Well, 42% said yes. 25 says partially. 32% no. So it's better you don't raise your hand. Oh, they're honest, yeah. And 4% is already familiar. That's probably one person in the room. Okay, thank you for being sincere, at least. But that's what we said, right? So it's going to always be a challenge when we try to do bioinformatics. And for bioinformatics, you need computational skills. So it's good. I mean, for the people who did the pre-material workshop, that's good, because you're going to learn more through the workshop if you already know what we're talking about. The way we build it, or we try to build it in a way that is going to be incremental difficulty. Well, that's the idea. Today, we're going to be reviewing part of the material that you have in those tutorials we're going to be with today. But after today, we're going to start assuming that you already know. So we'll see what happens. I think it's your turn, right? I hope I'm not carrying the microphone everywhere. Yeah.
Is this okay?
Yeah, I think it memorized whatever we typed.
Okay.
So.
exome or a genome or a targeted panel to find the cause of that disease, which could be a SNV in the genome of that individual. It could be a heterozygous or homozygous depending upon the mode of inheritance. And then from a research point of view, you could look at mechanisms of disease. Common diseases may also have some gene predispositions which you can discover by genomics. You can look for biomarkers for different diseases, that also you could do by genomics, and then you could amalgamate all that information to do something which is known as synthetic biology. But there are challenges in these omics as well. For example, there could be missing data in different levels, or there could be no data at all. So the solutions could be that you could discard those patients where the data is missing, or you could impute data from different omics layer to cover up for that data, but it may cause data distortion. For example, if you have mRNA data, you could impute that data to think about protein expression, or and so these are the challenges in multi-omics data. And this is just a timeline. I found this very interesting, of the timeline of different omics platforms. So I think Bonnie would be also talking about a bit of this history in his talk. So I'll just leave that. So the timeline for genomics, it started with the discovery of the DNA double helix in 1950s by Watson and Crick. Then the genetic code was deciphered. Sanger sequencing came in the late 60s and 70s. It was initially with radionucleotides, and then later it was automated with fluorescence. Then the PCR was developed by Carey Mullis, and then you had the genome project, which was a very ambitious project which was started and which was completed. And then you had next generation sequencing in the late 2000, and now you've got different platforms which are doing NGS. In epigenomics, you have the first discovery of histone methylation, then histone acetylation, DNA methylation, and so on. In transcriptomics, the initial part was with microarray, then you had RNA-seq, single cell RNA-seq, and now you're doing also spatial RNA-seq, which I think Bonnie would be covering. And in proteomics, MS and admin sequencing of proteins were the first technology, but now you've got high throughput platforms where you can do much more. And metabolomics also, you have got LCMS or MSMS based platforms. And lipidomics also, you've got lipid maps or other techniques. And then for all this data, you've got different tools for analysis. So with that, I think I would end here, and I'd like to think that it's like an elephant, and when you're looking at one particular omics technology, you're looking at one part of the elephant, but I think it's the whole which is more important. So I think next Bonnie would be talking about the basics of NGS and its applications. Over to you, Bonnie.
Quick question. I have a question for you.
Yeah.
Two options.
Yeah.
You can actually finish class like till 12 and then go for lunch. And then
No, lunch is from 1 to 2.
1 to 2. So,
Yeah.
so we'll finish top at 12 and then we have 1 hour
For the next
for
thing.
the next thing,
Yeah.
and then we go for lunch and then come back. Fine, fine, thanks. So I just wanted to know what is the hard stop there.
So it's 1, not
1.
12.
No, so 12 I will stop and then Francis will. Okay. So, great. So I'm going to talk for, actually this is a deck of slides, so don't worry about how many slides are there. I'm not going to really, maybe I will try to go through all the slides, but I'm not going to do that today, okay. It will be done over many days during the workshop. So, the idea that we should start thinking about is like what is actually genomics information or information encoded in the genome gives you. So, if you think about human to human variation, almost 99.9% of humans are similar in terms of their genome sequences. And interestingly, each of us has millions of SNPs also. So, which we don't know what those changes means. Okay. So, each of one are unique at one time, at the same time we are very similar also. So, which makes it very challenging for us to really make sense out of whatever variability what we are seeing. Okay. So, if you think what makes us us or what makes me is probably the 0.1% of the genome that actually makes me. And this really attribute it's into my color, hair color, eye color, height, everything else that actually comes with that. And this also related to drug responses. So, a lot of this differences what we see in human being and how they respond to drug, that actually also has to do with the small 1% variations that we have. Now, we cannot attribute everything to genes because it's too much. We can't blame or we can't give credit for anything good or bad happening to genes alone. It's actually gene and environment, that interaction, that gives you the final output what we end up being. And one example is this two fellows. One is Jim Fixx and other is Winston Churchill. Jim Fixx was an athlete. He used to train. He was a marathon runner. He used to train every day. Or you think about like marathon or runners, they have to have balanced diet and they will be eating all right thing, doing right stuff. But unfortunately, he died at age of 52. And Winston Churchill, he was a chronic drinker. He actually was a connoisseur of scotch and used to smoke a lot. And he ended up living for 90 years. So, which means that it's not just about your environment in which you are in, like you are training or something, but again the importance of genome in this process. What is the predisposition for you as a person, and when that predisposition interact with the environment means your lifestyle, your diet, your stress level, your general well-being. So, when that interact with the genomic information, but the genome gives the blueprint of that interaction. Somebody without a predisposition may not end up having, like, so you saw a lot of people actually talk about smoking. So, I can tell you one small story about my own father. My father started smoking at age of 17 and very recently he died. He died actually last year, and he died after he was diagnosed with COPD. But interesting story is this, which is part of the story is, he stopped smoking at age of 65, and that happened because my sister's son was born. He had a cardiac issue, so he stopped smoking for him, saying like he should not be exposed to smoke. So he stopped smoking for at 68, and after I think 3 years after that he was diagnosed with COPD, and he was in a ICU for some time, and then he was discharged. He came to the ward, and doctor came and asked, oh, you were almost about to die, and miraculously, he was miraculously saved. He was almost gone kind of. So the first, and then he said, did you smoke? So he answered, yeah, I started smoking from age of 17. And so he said, oh, okay. So doctor was actually not expecting the next sentence. And he said, everything was fine till I actually stopped smoking. So sometimes it's not, so this predisposition to a particular lifestyle issue is often important when we are thinking about the genetic component underlying those predispositions. So it's not just about environment, it's about the interaction of this environment. And so so to understand this more, we need to sequence DNA. And so what we are going to see today in some of these introductory talks, that we are going to look at history of DNA and sequencing. Then we also will.
We are not able to see the slides in the front. We have to see the slide or something. You don't have to see it.
But you can say it here.
Oh, I don't know. I can talk from here. That's not a problem. So yeah, we will be actually talking about applications. What are the applications of next-gen sequencing? Then we will also touch a little bit about pharmacogenomics and the variants, how we are actually dealing with those things. And then finally, we will be talking about single-cell sequencing, spatial analysis. Okay. So history of DNA actually don't start like in any recent time. Okay. It started way back in 1869 when the DNA was discovered, and people don't know that what is the nature of this DNA is. 1909, it was characterized, the chemical nature of DNA was characterized in 1909. And then a lot of things happened in between. People were trying to figure out that if DNA or RNA, what is the hereditary material, all these experiments were going on. And in 1952 comes the ability to understand that what are the real structure of this DNA. So the double helix structure of DNA that was given by Watson and Crick was proposed in 1953. And that was the biggest breakthrough in, and what actually it gave you, and the significance of this discovery was two. One, we understood the very chemical nature of DNA and its structure. Also, we got ability to manipulate DNA. So the outcome of this discovery was, which later became recombinant DNA technology, where you can cut and chop DNA and make a new hybrid molecule. You can do so many things with that. And what also it led to, it actually converted biology from a descriptional science to an experimental science. That means you now can actually manipulate the biological system, and you can measure the outcome of that in terms of system, like what is happening to those manipulation, what is the effect of those manipulation on at the system level. You can do those measurements. And then came 1977 when Sanger sequencing was discovered. And so at the same time, there was another platform or other way of sequencing came, which is called as Maxim and Gilbert. And the reason why Maxim and Gilbert was not that popular, or also not now also popular, because it was not automation friendly. So Sanger sequencing was automation friendly. So the first genome was sequenced, it's a bacteriophage genome, which was just 5 kb big, using Sanger sequencing in 1977. And first automated Sanger sequencing machine came in 1986. Interestingly, after all these advancements, still these machines are actually operational in labs, including one in Yale. So people are still doing, for low throughput sequencing, people are using Sanger sequencing as a means to sequence. Okay, and in 1990, people just thought that, okay, let's sequence genome. That was the beginning of Human Genome Project. So it was not like people just started sequencing human genome, because it's a big genome. People were sequencing 5 kb, 10 kb genomes, but they were not used to sequence a 3.5 gig genomes. So a lot of background work needs to be done before really they start sequencing human genome. So first what they did, they started in Baylor College of Medicine, they first started a sequencing factory, and they sequenced a bigger genome, which was a bacterial genome for influenza, which was around 1.8 MB long. So that is when they established all the ground rules for aligning, stitching, and back library preparation. And 1998, they sequenced first animal genome, which was a nematode from C. elegans. So it's a nematode, and its genome size was 97 MB. So now you are seeing the first one was 5 kb, then they sequenced 1.9 MB, then they sequenced a 97 MB big genome. From there, they started working on human genome, and in 2003, they completed human genome, which was about 3 GB of sequence. So it's a big, huge genome, and it took almost 13 years and $2.3 billion. Okay, now I have to give you a perspective. I don't know how many of you are familiar of billion word. So million is 10 lakh, okay, and billion is 1,000 lakh. So that much dollar, and dollar is around 83 rupees, and now it's actually 86 rupees per dollar. So that is a huge amount. So people have spent that much. Then in 2000, so one of the outcome of human genome sequence was not just about sequencing human, but it also bring in new technologies for sequencing. And the result of those new advancement was the development of this new technological breakthroughs called as next generation sequencing. It is just because they were using Sangers and they wanted to give a name, so they gave it next generation sequencing. So this is an umbrella term that is used, but the whole important aspect here is that one side they could actually sequence genome, but they also developed new ways of sequencing, cheaper way of sequencing genome. So please be keeping this in mind, 13 years and 2.7 billion, because we will be visiting this number shortly. So at this point, what is really happening is that a large number of human genomes are being sequenced. In Sanger's Institute, they just completed exome sequencing of 243,633 genomes. All of Us program, which is a US program, they just completed 250,000 genomes, and their target is to sequence 1 million genome. UK Biobank has another 500,000 genomes, and they just released 450,000 genomes, whole genome sequencing. So, all these are exome sequences, but the UK just released their whole genome data set, which is around 450,000, like 450,000 genomes. So, lot of high throughput sequencing projects are going on all around the world, and UK also just launched, they called England newborn screening or something, which basically idea is that every single newborn baby in UK will be sequenced, and their data will go into their clinical records, and clinicians will use that for their future. Anytime they want to use that information for any kind of interpretation, they will be using that. So, this is going on, and we are also currently working on a pilot project where in US we are doing this similar newborn screening program in a primary healthcare setup. So, we haven't got grants yet, but we just wrote that a grant proposal for them. So, it's not just human, because this planet belongs to other animals too. So, a large amount of animals from every single phylum is being sequenced now. So, a large scale plant and various animals are being sequenced at this point of time, and people are using sequencing as a tool to study various aspects of biology. So, one of the key advancement that is happening at this point is that the sequencing, there is a war going between the companies for the sequencing companies to slash down the cost of sequencing, and that leads to various platforms which gives you a very cheap sequencing. And at this point there is a platform called as Ultima. They just released $80 genome. That means you can do less than $100 genome, whole genome for human. And so which is like unheard of. So the number that we were talking about, you remember, $2.3 billion, coming from $2.3 billion, now we are talking about $100 genome. So $100 genome, even if you think from Indian context, if 8,000 rupees you have to pay for a clinical test, everybody will be ready to pay for that. Is that right? So which means now, because of this cost of sequencing, the sequencing is now possible under the patient care setup. Because if you have to pay, like say $1,000 for a genome, not many insurance people will be ready to pay that. And if you have to shell out that kind of money from your pocket, it's a big amount. But $100, yes, I'm happy to pay for, $100 for that. So the whole idea is to give a $100 genome in a clinical setting, so that people can use, more and more clinicians, more and more people can use this genome sequencing as a means for various kind of diagnostic purposes. Okay, so this is really getting real nowadays. So cost of sequencing, as I mentioned, that this is dropping down significantly, and most of this is happening because of the invention of new sequences, and new companies are actually doing a different ways of sequencing it, and we will see in coming slides like how various companies and various platforms are using sequencing in different ways. So this is actually crashing the cost. Meanwhile, what we are at this point is around $350 genome for a 30x whole genome sequence for $350. So this is the price that we are charging. So compared to $2.3 billion, and this happens within 48 hours. That is another thing. And very recently, I am also going to talk about that sequencer, which is released by Roche, and they have shown that they can create a whole genome sequence in 4.5 hours. So not just whole genome sequence with a clinical report can be generated in 4 hours and 30 minutes, and this is a world record now. So they just got the Guinness. And last week I was in Boston ASHG, and where they released that information. So we are actually talking not just about the cost here. So $350 or $400 genome we are talking about, but at the same time we are able to generate data in 4.5 hours. And I mentioned about rapid whole genome sequencing, which we are generating data to sequence sequence to data within 30 hours. So which means a NICU PICU kid who is waiting for a diagnosis will be immensely benefited if you have a high-speed sequencing platforms and at a reasonable cost. So cost is also, because if you are spending $1 million to gain something, it's not useful. But then with such a cost, it's actually very much useful. So sequencing history is divided into generations. So we talk about, the first generation sequences is essentially Sanger sequencing. So the first generation sequencing is based on, there were 4 different innovations were there in the first generation sequencing. The first was chain termination method, which was Sanger's method, which was later on automated. Then you have chain degradation method, which was Maxim and Gilbert method. Unfortunately, this was a very robust method, but it's hard to automate, so it could not get much traction. Then there are two other methods. One is called as pyrophosphate sequencing, and second is ligation enzyme method. So all these four methods were developed during first generation sequencing, but the last two, pyrophosphate sequencing and ligation methods, were not commercialized till second generation of sequences. So only the first generation sequences were using chain termination methods. Okay, and one of the major advantage of first generation sequences were its accuracy. It was highly accurate. Highly accurate means like highly accurate. Very rarely you have a chance like you will get a wrong base calls on them. But the problem is you are sequencing one molecule at a time. So cost of sequencing is very high and throughput is very low. And one of the example for, as I mentioned, for first generation sequencing is Sanger sequencing. So here we are actually what we are doing. We are doing something called as chain termination methods. You will mix normal NTPs along with the D-deoxy NTP. So whenever a D-deoxy NTPs are incorporated, it will terminate the chain. Okay, and each of the D-deoxy NTPs are labeled with one unique color fluorophore. So that means you will generate a series of fragments of various size wherever the termination is happening, and then when you run an electrophoresis gel, and then you can read the color from top to bottom, and you can deconvolute the sequence, the order of those sequences. So because each of the fragments are actually stacking at different level, and these colors are read through a laser. So basically electrophoresis is happening, laser is at the bottom, and it will, for the first base will pass, which is the shortest corresponding to shortest fragment. So that will be the first base. So similarly laser will read that, and the second base will pass, third will pass, fourth will pass, like that it will pass. It will create something called as chromatogram. So this is how actually it looks like. The chromatogram, you can actually deconvolute this into a sequence, and the chromatograms can be converted to sequence. So this is what actually, which is what done in the for the first generation sequencing using Sanger sequencing. Okay. Now, as I mentioned, like once they have done with the Sanger sequencing, now it comes next generation sequencing. It just means like you created a next generation sequencer. So next generation sequencing can be divided into basically 3 different generations, and I just mentioned 2 here because of one particular reason. I will tell you why. So the second generation sequencing and the third generation sequencers. Second generation sequencing were short read sequences. So you will fragment your DNA to short fragments and sequence millions of molecules in parallel, and then you align it back into that, and you all are familiar with the short read sequences. And the technologies they were using for short read sequencing were either sequencing by synthesis or sequencing by ligation. You remember I mentioned like first generation sequencing, they developed this sequencing by ligation, but the real use of that technology came only in the second generation of sequencing. Okay. Then came the third generation of sequences where people were using long read sequencing technology. So instead of using short fragments, they started sequencing bigger molecule, and that you can do using either single molecule sequencing or something called as synthetic long read. So synthetic long reads are not something used anymore. So but then most of the we will be looking at other long read platform. I mentioned like there is another fourth generation of sequencers also there, but essentially they are not based on sequencers, but rather than they based on approaches. So like all these single cell studies or the spatial technology where they are using microfluidics and other means to make a novel library prep method. So it's essentially the new library prep method was driving the fourth generation sequencing technology. So which is single cell technology or spatial technology. So sequencers were not generated, the new sequencer did not come during this period, fourth generation, but new library prep methods came which allowed you to look at genome at single cell level. Okay, that was the fourth generation of sequencing. So let's look at second generation. So I know most of you are aware of all these terminologies, but just for I was not sure about what is the level of audience. But based on your introduction, it seems like you all know these things. But just to summarize quickly, so what is a read? Read is basically a inferred sequence of bases corresponding to a single DNA fragment. So as I told you, like we are sequencing millions of DNA in parallel. So each molecule, the sequence originating from each molecule is actually called as a read. Okay. Now I have a question for if anybody could actually give a stab to it. So my question is, why I'm using this word called as inferred? No, that has nothing to do. I'm inferring. I'm not actually saying. So in case of Sanger sequencing, I was super confident. I said that this is a sequence.
It is not exactly by, so it occurs by fluorescence or by ions or something like that. So the sequence is read as a analysis.
Yeah, I know what you're saying. Yeah.
The reader of a signal.
Yeah, essentially there is a third party software or vendor software which is telling us that this is the base. In case of Sanger sequencing, we exactly know that this is the base there, okay? But in case of this, we are inferring, okay? And so that is why there's always an ambiguity, like whether this base is right or wrong, okay? So that is why that's why it's not called as a true sequence, but rather than it's called as an inferred sequence. And the quality of sequences are so good nowadays that this really we don't have to use that definition anymore because they are extremely accurate. But still, for a historical context, it's always good to have that definition as a part of our discussion. Now, what is actually?
So what do you mean by read? Is the typical sequence? It
Yeah.
could be 10-base sequence.
Yeah, it can be any base sequence, but it should be originating from one single molecule. So you are sequencing millions of molecules in parallel, and each molecule will give you one set of signals. So if you are sequencing say 150 base pair, so you will get 150 base pair reads or sequence from each of this million molecule. Now each of them are called as a read. Okay. So now other thing is library. So those who are in genomics, you are familiar with this term library. So let me ask you, when you think about library, what comes in your mind?
Many things that
Yeah.
whole
Collection. So collection, okay, that's the first thing. So what is the nature of collection? Like, is it same book of 1,000 copy or different book of, what is actually collection means?
It's a mixture.
Mixture. So it's mixture. It can be one copy, it can be 5 copies. Some of the books may have 100 copies. Some of them will be rare manuscript and you are not supposed to touch it. All these things. Okay. So the library is actually collection of molecules or collection. So when you have a collection of DNA and you want to sequence them, then you add certain things at the end of it, and that actually that is important for making sequencing. And once the molecule is ready for sequencing, those molecules are called as libraries, and they have same origin or have different origin, they have different molecules, different sizes, everything. Okay. Now the third idea is called as coverage or depth. So this is a very important question when you are doing an any NGS experiment. The first question or two most important questions that you should be asking: how many replicates I need, and what coverage I need. Coverage means how many times a base is covered during sequencing, and why you need to do that.
accuracy. Accurately we can determine the sequence.
Yes. So one of the example I can tell you, like if we are, so suppose if I have to tell you that go from here to say rock garden and document everything that you see in during your way. If you go once and then come back, then what will happen? That you may miss something. But if you are going 30 times, the chances like you will cover all the landmarks and all the information. So the coverage is very important, and depends upon how rare the event that you want to capture, you will have a deeper coverage you need. Okay. And definitely we also need to learn about flat form and how you calculate coverage. Coverage is actually calculated through, okay, thank you. So coverage is calculated through the number of reads you want, you multiply that with what is the read length. So how long you are sequencing those ones, and then you divide that by genome size. So if you are looking at RNA-seq data, then instead of genome size, you are looking at transcriptome size. Okay. So genome size versus transcriptome size. Okay, that's. No, that's fine. Okay. So basically you take the number of reads you have, you go for the what is the length of the reads. So suppose if you are doing a paired end reads, then you multiply this by 2 and then divide that with the genome size, and if it is a transcriptome, then divide that with transcriptome. Okay, that is. Okay. Now sequencing can be done as a single end sequencing or a paired end sequencing. Okay. So single end sequencing means a molecule can land either side, like 5' end or 3' end, and sequencer will go from one end to other end. By paired end sequencing, so sequencing will be done from one end to other end and then flip and then sequence from this end to this end. The idea is same what I was mentioning about our trip to rock garden. If you go once and just stop there and then come back, you never know that if you are seeing a difference in the bases, is it real or is it a error during sequencing. Now when it comes to error, the one of the main feature of error is errors are always randomly distributed. So same error will not happen same place same time twice, it's always randomly distributed. So if you actually sequence same molecule twice from one end to other end and you see the same changes happening in the base, then you can ascertain that those changes are real. But if not, then then you know that there is some issue, and then you can correct it for those, and there are ways to do that bioinformatics. Okay. So now when I started as a postdoc, that time sequencing was very expensive thing to do. I'm talking about $100 genome, $350 genome and all. But when I was doing, it was like if I have to sequence a genome, it will cost me around $10,000 to $12,000. It was 15 years back when I was a postdoc. So and then paired end sequencing was like further expensive. But nowadays the paired end sequencing and single end sequencing costs are almost same. So we are not offering any more single end sequencing because it's not worth offering single end sequencing. So most of those earlier consideration that whether you will choose a paired end sequencing or single end sequencing was purely based on necessity of the application. Some of the application where you need extremely correct reads, you will be actually using paired end, otherwise you will use single end because the cost was so much different. But nowadays, since the paired end sequencing costs are almost, or some cases actually it is cheaper than single end, so essentially we are using paired end sequencing for all of our applications at YCG. Okay. Now let's talk a little bit more about libraries, the kind of libraries that we make. So libraries can be directional libraries, it can be non-directional libraries, it can be targeted capture libraries or amplicon libraries. So the directional libraries where you maintain the direction of the molecule, and where you need such an application. Can somebody guess where we need where we need to preserve the direction of a molecule? Anybody? Probably not plasmid. You are not going to sequence whole plasmid, no. Where do you think you need which molecule has a directionality in genomics? Yes, maybe, but then where else? RNA is where you have the directionality. Okay, but again, there's another issue there with the RNA. You can always align RNA to a sequence, and then you can still find the directionality. Then why you need a directional library? No, it will, but then from a practical standpoint, if you can align your transcriptome back into the transcriptome database, you can still align it back, and then you say, okay, this is the thing, and then you can quantify whatever you want to do. Nowhere actually you read, in that case you don't need a direction, you don't need to know the directionality of it. Okay, but anyway, so let me actually give you some insight like why we need some of those directional libraries. So there are transcription, the way we think, especially in mammals, vertebrates, it's not that simple. There are a lot of transcription happens within a gene or from a So like a lot of genes are actually transcribed from the same genomic location in either direction. So there are divergent transcriptions are there are actually convergent transcriptions are there are transcript originating within a transcripts are there, the start sites are actually different from the same gene. So all these places are there where you can use the information related if you preserve the direction, because bioinformatics won't give you the directionality in real terms. So if you can preserve your direction, that means you can make more sense out of those mechanistic things. So for a normal bulk RNA-seq where you are just looking for differential gene expression, directionality is not that important. But if you are looking more into mechanistic details, you are more looking into those genes which are transcribed from a single loci or non-coding transcription. Some of them actually originate within the intronic regions or within the gene, and microRNA. All those areas actually directionality helps you to understand that data a little more. Okay, so you make directional libraries, and most of the cases directional libraries are made for RNA. Now targeted capture, a lot of you are working on that, exome libraries, or you can have a custom panel to capture it. Then you can also amplify PCR amplify regions, and then you can sequence them using NGS using amplicon libraries. Okay. So one of the key idea of sequencing, and you will be seeing some of this when today afternoon when Francis will be going to talk to you about FastQC, that the Q scores. Okay, so sequencing quality is given in terms of Q scores, and Q score stands for 10 to the log 10 E, where E is the estimated probability of incorrect base. So basically, you are looking at what is the probability that a base called by your base caller is incorrect, and if it is Q10, which means every one base called from out of 10 will be wrong, which is like really we are sequencing a million base, that means you are almost 100,000 bases, 1 lakh base will be wrong. Okay, Q20 means like 1 in 100, which is around 99% accuracy, and Q30 means 1 in 10,000. So the new sequencers, so Illumina sequencer operates at a level of Q30, that means 1 in 1000 base error. There is a new sequencer called as AVITI. The sequencing quality is in Q40, which is 1 in 10,000, and they also came up with a new chemistry which can give you Q50 read value, which is 1 in 100,000 base error. So which is extremely accurate sequencing done by these platforms. Okay. So NGS platforms, when you think about platforms, there are various platforms available and they differ in chemistries. And then one of the key characters of NGS or next generation sequencing, that they can sequence millions of clonally amplified molecules in a massively parallel manner. So this is the difference between Sanger sequencing or first generation sequencing. So two things, that you should be able to sequence millions of molecules and in parallel, which means every single molecule is sequenced along with others. Okay, and instead of doing this gene by gene, you can actually do everything together. So what actually really differs in case of various sequences is that how you do clonal amplification and how do you reaction, like how you measure the output of that and how do you do the clonal amplification. Now, can you tell me why we need to do clonal amplification? Why we need to amplify that signal? Why we need to do that?
Accurately, we can determine the sequence if there are more number of molecules, more number of copies of that molecule.
Why?
More number of times it is read. More number of times the thing is read again and again.
No. So this is the clonal amplification. Clonal amplification actually is defined by in a sequencer you load one molecule or say one spot you have, and on that spot itself it amplifies and it actually creates a bundle of fragments there.
So all the signals should be, so for base accuracy,
Signal
it's very
amplification.
important that the fragments is.
Signal amplification.
Yeah, so signal amplification. So signal to you want to achieve a high signal to noise ratio. So here is my question. Then why don't we use a very bright floor? So instead of amplifying signals from a multiple floors in a bundle, and get a very high signal to noise ratio, just put a very bright fluor, which is really bright in a dark background. That means you will be able to get it.
Sometimes wrong bases are called. So
No, I'm fine with that. I'm sequencing millions of molecules. I'm fine with like 1 in 100,000 molecules being called, 1 in 100,000 bases being called wrong. I'm fine with that.
Some genes are very low in copy number. Some genes have high copy
No,
number.
it has nothing to do. You are not talking about gene here. We are talking about fragment. Each fragment we are amplifying. We are keeping a bundle. And I agree with your idea that we are actually doing that to increase the signal. And then that way we are actually increasing signal to noise ratio. So we are reducing the noise and we are increasing the signal. So now it's like night sky. You are actually seeing a brighter star. Okay. So my question is, rather than doing it, so in this case you are using a dim fluor and you are using multiple dim fluor and you are aggregating all the signals together to create a enough bright signal, and then you are capturing it. But instead of that, why don't we actually put one single bright fluor and say like, okay, I'm done with this, I don't want to do clonal amplification.
I just want to guess. Do we have a fluorochrome like that, number one? And number two is the spectra varies, overlapping with the other colors maybe.
We have, believe me, we have a fluor like that. Okay, we have a fluor, and if not, then somebody can make a fluor for that. Hmm? Okay, hold on to that thought. Okay, sensitivity. Then anybody else? Okay, so let me actually
So
give.
signal will be too bright that we may have problem in reading that signal.
Yeah, that is a problem. So there are two problems there. If you have, so how this technology works actually? There's a high-speed camera. It's actually taking pictures. Okay, so if you have a very bright fluor, then what will happen? The two molecules, they are actually near to each other, the signals will merge. Then the camera won't be able to resolve it, which means like you won't be able to sequence millions of molecules. Rather than now you have to reduce the number of molecules and some way, and you have to space it enough, okay, so that those signals can be. But there is another bigger challenge for also. This is a technical challenge, but there is another financial challenge. When you actually employ a brighter fluor, the sequencing reaction will become more expensive. So it's not that we don't have brighter fluor, we have brighter fluors, but the cost of sequencing will be dramatically high. So that means that both the things, like you want to control over the cost of sequencing reaction, at the same time you want to make it as dense as possible. Like I was talking about NovoSeq X Plus platform, in single flow cell, this is such a small flow cell, and it can sequence up to 25 billion reads. That means it's such a dense environment that the molecule to molecule you don't have any much space left, and if you have a bright fluor, then it will blind everything, and then you won't be able to resolve it at all. Okay, so the most important thing, right, that you cannot resolve it, and then cost is actually very high when you are using bright. So we clonally amplify. Okay, now for the pyro sequencing you use emulsion PCR. Same for the the sequence mobilization also use emulsion PCR, and then ion torrent you also use emulsion PCR, while for Illumina based sequencing you use cluster generation, and we will be seeing this process in a little more detail. And the output is captured. For the pyro sequencing we will be using luminescence, while for solid we are using fluorescence. Ion torrent we are using pH change, and then for the fluorescence based imaging we are doing for the okay. Well, yeah, 10 minutes. Okay, so let's start with some short read sequencing platforms. The first one what we are going to look at is ion torrent. Okay, so ion torrent actually senses pH change. Now how it is, what it does, you have a DNA, and then you have a primer. Actually for the sequencing primer you add the sequencing primer, and sequencing primer will go and align to this. Okay, after that you will flow your first base. So you are here, actually you are adding one nucleotide at a time. So you will add your first base, and then you try to measure that if anything happens in the pH. So if there is a complementary base pairing, then there will be a release of an H plus ion, and that will change the pH of this micro well. And that change is actually sensed by a pH sensing layer, And the moment that pH change is there, it will register that, oh, once you flowed, if the flow, this particular base was flowed, you have that pH change, and then you register that as that base. So like in this case, as soon as T, if A comes, nothing will happen. There's no chemistry. Kuch kuch hota hai type. If you don't have kuch kuch hota hai, then you will not see that. So the moment T comes, then you say, okay, kuch kuch hota hai, and then you sense that kuch kuch hota hai in the pH layer, and then you will. So this is through sensing that H change in the ionic concentration, H plus ion concentration in that local environment, and that will give you, and that is how you are doing the sequencing. Okay.
There will be always change in one edge, right? So how will it differentiate between the bases? So when there is a complementary reaction, though there will always be the same change in pH, or there will be variation?
No, there is the same change. So what happens usually that you flow first base. So that is what I was saying, that you suppose in this case the first base is A. Okay. So you say the first base came inside the flow where is C. C, nothing will happen because there is no base complementarity. So, and then you wash off the C, everything from the well. Then you put second one, then the third one. So whenever the real complementary base comes, the H plus ion the change will happen, that will be sensed. Then you wash off that reaction. Now the well is back to its original state, but at that point you have the first base already reverse complement. Now you are looking for the second base. So now you are looking for G to G. So like that, it goes and it does in.
Depends upon we know the flow of the bases what we are throwing at them.
Yes.
In relation to the flow of
The nucleotide.
coming in.
Yeah, the basis which is coming in. This is a very simplistic view of ion torrent reaction. There is certain primer based jugglery is also been done in this, but the basic idea is this: like base comes in, if it is actually a complementary to to the base that is there, it will release a H plus ion, and that H plus ion will be sensed by this pH sensing layer, and that.
But the only problem would be when there is a string of same nuclei.
Yeah.
Homopolymer repeats, because there are a lot of string of A's or string of G's. Then to differentiate that, how much of that is because of 4 or 5 or 6 or 7 becomes good. And so this kind of chemistry is very poor for regions where there are homopolymers.
But unfortunately or fortunately, this is one of the most widely used platform for clinical application. A lot of faster, I think. Yeah, clinical setup. You see a lot of ion torrents working in clinical setup. Okay. The other platform is called as nanostring. Okay, so it's encounter platform. This is basically this is a counting platform. So you are not sequencing here. You are just counting. And what are you doing here? So suppose this is your so it has each gene is actually characterized by 2 sets of probes. One is called as capture probe, and another one is called as reporter probe. And reporter probe will have a set of fluorescent barcode. At each of the reporter probe will have a series of reporter barcode defining each of those genes. When you hybridize, then the capture probe and the reporter probe will go and hybridize to the gene. So now the one side is for pulling it out, and second side is for reporting. So unless you have both the sides on the same molecule, you will not be able to do anything. So once the right gene is there, then that means the capture probe and the reporter probe will go and hybridize to the gene of interest, and now you wash off anything that is not hybridized, and that means you are left with a series of hybridized molecule with a distinct combinatorial fluorescent makeup, and the reader will go and read and just count like how many of them are there. That's it. It will just count like, okay, this particular combination of fluorophores, this many number, this, and then that you just convert that into name of a gene, and this is how and then you end up having a data something like this. So here you are not sequencing, but rather than what you are doing, you are just counting. You are just counting, through hybridization. And
So it's about the gene expression.
yeah, it is mostly used for gene expression analysis. So again, this is one of the platform that has a lot of traction in clinical setup, because you can make small panels like 60 gene, 80 gene panel, and instead of doing RNA-seq and then doing all this complex analysis, you can quickly get the results out of those clinical samples. If there is an assay you can develop on this. A lot of people actually love to do this for cancer and other area, but this is again because of the simplicity, and the only issue with this platform is that initial cost of setting up this, because those synthesis for this probe is expensive. So once you synthesized it, it will be for hundreds and thousands of reaction. That means per reaction cost is low. But if you have only few samples, say I have only 10 samples, probably I will not go for this kind of panel. But if you are going to use a panel for repeatedly for your clinical application or in your lab, say like your rest of your 5 years you are going to do thousands of samples, then this is the cheapest way to do a gene expression analysis, and you don't need any bioinformatics support to do so. Okay.
Besides cancer panels, like what other applications this can cover
No,
besides cancer?
no, everywhere, wherever you have a gene expression. Gene expression. Yeah, and like best example is infections. So like you have a RSV panel, or you have a respiratory virus panel, 200 virus panel you have. You just create a set of like probes and reporter gene for each of them and run it. Like within a minute you will be able to quantify. And the good thing about it, it's sensitivity. Even for single virus level, sensitivity it can give you with those. But again, the main issue with encounters are that unless you have enough number of sample to analyze, it's not worth, because of the initial cost of generating the probes and reporters are actually high. But if you have enough number of samples and you are going to use that for over years, then I think this is a great platform.
What is the advantage of this over droplet digital PCR?
Droplet digital PCR, so two things. One, droplet digital PCR is still very low throughput. You cannot, I mean, most of the platforms cannot go, I believe Coagility is the only one which can go up to 12 floors. But most of them are between 4 to 5 floors. So that means you cannot analyze more than 5 genes in a droplet digital PCR. And it's expensive. The reagents are expensive. The Tachman probes are expensive. Then the CyberGreen is expensive. Everything is expensive there. So that is what I was saying. If you have only few samples, say like 10 samples, probably digital droplet PCRs can work. But if you have enough number of samples, and then if you're looking for 60 or 80 or 100 genes, then the encounters are the one. For larger number of genes, encounters are, droplet PCR good for like fewer genes. And if you have a larger number of sample, again encounters is that one.
So reported dyes of multiple fluorescence?
Yeah, they actually use what they call, what is that? Quantum dots.
Quantum.
Yeah, so when I was actually presenting, I was thinking of that term, and I was not getting it. So thank you for asking me. So they use quantum dots, and then they actually create a combination, and then that is what they read.
So each reported dye will have for each gene, or they will give a
Yeah.
combination of?
So yeah, they give a combination of quantum dots, and that combination is unique. And the encounter, the instrument reads that combination and deconvolute back into the gene of interest.
This encounter technology, is it, IVD approved for breast cancer?
I'm not sure about the IVD approval, but in US there is a way we actually called as LDT, lab developed test, and which can be, so basically the difference between IVD and LDT is that LDTs are usually done by individual labs. So like my lab, I can develop an LDT and I can validate those LDT based on CLIA cap regulation guidelines, and then I can offer that service to anybody I want. But that cannot be used outside my lab, because it is a protocol, a assay that is validated just for my lab. But IVD is something that you do a large test, you file it with FDA. FDA will give you approval, and that means once you have an IVD approved, that means anybody can use that technology by paying a fees to you. So I'm not sure about IVD part, but I know that a lot of labs are actually using this under LDT licenses. Okay. Now, just before, okay, I think I will stop here probably, and then we'll have tea. And if we are left with like 10, if you come for last 15 minutes, I will use probably, or you can actually take it from there after tea. No, you start, and I will continue next like day or so.
So we're going to have a break now?
Yeah, they will have a tea break for 15 minutes, and then you can set your things up, and then we can go. Okay.
The tea break would be downstairs, outside of spaces where we
Can you pass the slides? No, right? But we can plug it here, and then we can pass the slides.
Okay. Can you try that?
Right? So if we plug in here, we can pass the slides with this. All right, good. Thank you. Is the mic working? Yes, good. I'm not going to sing, so you're safe. I'm going to try. I mean, I haven't figured out yet. Oh, it was working a moment ago. Oh, now. So again, we're going to try again. It doesn't work. I don't know why. It doesn't show the results live. But I'll try something different now and see if I can make it work. You saved the address, right? So I don't. Okay. Come on, you work a moment ago. Oh, now. Okay, good. No, okay, it's not working. We'll skip it.
यहाँ पे आ रहा है. It's coming on our screen.
Oh, the question is coming in your screen? Okay. Yeah, so the next, so if you go to polev at that address, FLOPZ048, I can put it back on. Let me see. Oh, I need to point there, right? I don't know why I'm pointing to the screen. You know what? I'll just sit. So if you go to this address, you remember it? Okay. So I think you're going to get a question, something like, what bioinformatics tools are you aware of? So if you don't mind typing what tools you are aware of. Any. Pick one. And so the trick though, if it's a multiple word, don't use white spaces. Use an underscore to connect the words.
We have heard only the name. We are not actually aware of it.
You know any tool? Just write down the tool, the name of the tool. Anything that comes to your mind. What tools you are aware of? Yeah, so if you're going to type bow tie separately, you need to use the underscore. So I can see somebody mention Trimomatic, FastQC, Picard Tools, Anovar, SAM Tools, some databases like CleanBar, HiSat2. So everything quite focused on alignment, annotation, R, BLAST, okay, Python. Yeah, I don't know if it's going to be readable. Yeah, more or less to give an idea, right? So yeah, you give a lot of good ideas, right? All these are programs that we use for analysis, even databases, some tools for visualization like IGV. I can see annotation tools here. So we need to define bioinformatics, right? The first thing I did, I went to the Wikipedia. Sorry, I'm in the middle again, right? I went to the Wikipedia, and one of the things that first shocked me, right? So it mentions that it's an interdisciplinary science, which is true, right? I mean, maybe not in the tools that we were talking about. All of them were about genomics and annotation, a lot of alignment tools and things like that. But yeah, definitely bioinformatics includes programming, math, statistics. Somebody was mentioning R, definitely. Nobody mentioned anything related, I think, about protein structures, but there's bioinformatics in a lot of different fields. It was there? Okay.
No, I don't think it was there.
Oh, okay. Yeah, yeah. AlphaFold, all those tools, right? Creation of algorithms. So math is there's a lot of involvement of math too.
But have you come across any errors? Because recently we found that for some particular diseases it was calling variants wrongly.
Which one, sorry? Oh, the one from Google. Yeah, I don't know.
No, for like FMF,
Okay.
familial fever. So some of the MEFE variant it was calling benign, which were clearly pathogenic. And alpha missense was calling them benign.
Yep. So yeah, quite interdisciplinary, right? I think all of us could agree on this. So that has an implication, definitely, which is the skills required to be a bioinformatician. If you need to match all the fields, it's going to be complicated. But definitely, there's a long list. And here, probably, I'm just listing whatever come to my mind at that moment, right? So programming. As you can imagine, you're going to need it at some point. Sooner or later, you're going to need to learn some programming skills. Definitely, you need the statistical thinking, data management, biological interpretation, and things that you probably don't think that much, but data ethics and privacy, it's also involved, right? When you work with bioinformatics, especially if you work in a clinical setting. And nowadays, it's very difficult not to talk about machine learning when you work in bioinformatics. So another thing you probably noticed, right? So when we talk about bioinformatics, we talk big files, a lot of sequences, and things like that. And generally, a laptop, it may not be enough to do what we do. What we would generally do is generally use the laptop as a Windows to connect to a bigger server, right? Working in a cloud environment, or in this case, this is a picture of I think this is the old one, actually. This is a very old picture, but the cluster that we use at the Genome Center. This was the old cluster that we use at the Genome Center. But it also exists other type of clusters that you connect to.
In your center, how much of data is stored in your servers? Like petabytes or like?
So I don't know how much we have stored. I can tell you we store everything since 2009. But
a lot of data.
Yes, a lot of data. The funny thing is I remember like the first year we used to tell to the user, oh, we're going to erase the data in 1 year. So it's better you figure out a way to store it and something like that. That wasn't true until 2016, after a few years. And then we start removing them from the actual cluster, but we have a backup. We used to have a backup tape. So the old classical magnetic tape, because it was really cheap, we stored the data on a tape system. Nowadays, I'm not sure, but I remember, I don't know if Bonnie knows. Bonnie knows more the economics of the center.
Is it economical to say RAM or CRAM, or what is economical to say?
We store the raw files. We didn't store FASTQ files. We never store anything processed.
Are they bigger?
Yeah, they're probably bigger. We could use CRAM and store the CRAM and then transform it back to FASTQ if you need to.
Yeah.
But no, we from the beginning we start storing FASTQ and that's where we stay.
One thing actually you should think about when you are thinking, one of the biggest challenge for genomics at this point is data storage. Okay, because we are creating data at a speed that we don't have that much ability to store. So archiving, and between this CRAM formats or any other compression formats, the biggest challenge is the downstream pipeline needs to be accommodated, like CRAM files. They're actually Lumina format. That means you need to actually have some way of decompressing it, and that means you need to pay something for that, those kind of things. So when you're storing that in a certain format, you also should think about, is your downstream pipelines are compatible with your compression, and what it will cost you to decompress it. So compression in CRAM compression from Illumina is free.
I'm blocking.
Decompression you have to pay money.
I'm good, or I'm blocking?
So that is yeah. Clouds and everything is paid, but then those are the things that you're really thinking about,
like moving like this.
how often you are going to download the data, because most of the cloud will have very limited amount of money for very limited cost for uploading. The cost is in download, so you are frequently downloading, then you are going to spend a lot of money.
If we want to store the human genome data, and how much we can store it?
So usually what we do, we store data only for 6 months, and then we archive it in the tape.
Yeah.
So we move it to the tape archival system,
I think the tape is gone. I think we will use now a different system. But I remember somebody, I mean, the cost, I remember at some point the cost only of all data saved, it was $10,000 per month.
Yeah.
And then we realized.
So isn't it good enough that if you want to sequence it, resequence it rather than store it? So there's now a lot of talk that rather than storing, which is so costlier, you might as well resequence it.
Store the DNA and.
But the problem, I mean, see, if you are working with a research sample, you will have enough sample to do that. But most of the data
individual is there,
sample do have that much.
But it might change if you are looking for some inheritance.
If it is temporal, like it has changed over
Yes.
time, then probably.
We still go back to data in 2012.
Yeah.
So,
Storing is still.
so we need to store it because people is asking for data from as old as 2012.
And there was some talk about, I don't know how far it has gone, that the best way to store data is like how it is stored in the DNA itself.
Okay, so I should tell you one
Like,
story.
so there was some project
I work with it.
where they're trying to store
The picture of.
data from the genome in the format of a genome itself.
Yeah.
So I will tell you one story. You have that picture of you holding that capsule. You had to take it.
Oh, I don't know what that picture is.
This is an interesting thing Dr. mentioned. So Francis was actually involved in a project where two novels written books were stored in a capsule in a DNA format. So what they were doing, they were actually taking. So this is the lifelike DNA. You add water, you make library sequencing, and then stitch it back, and it will give you a novel protein.
Well, there's a conversion system, right? So the DNA would give you back
And so
the book.
Francis was analyzing that, like he was creating that book back, I remember. And he had a picture, I still remember, holding that capsule in his hand.
How big were those novels?
Oh, you can store a lot of it.
War and Peace of Leo Tolstoy?
No, I don't remember what books were. I completely forgot.
We did it back in 2022, and this was the first time Swiss biosciences were doing it. And now this idea is new because of, they had a way of making, so one of the costliest things, cost issue in this approach, like when you say something in DNA format, is the cost of oligosynthesis. So Chris came with an idea for that instead of sequencing, in making individual oligos, they do something called pooled oligosynthesis. That is, they create a pool of oligo on a slide. So that reduces the cost. And so that is why they were testing the storage one. And they reached out to Francis for that, and then we did the sequencing and Francis did the analysis.
So you will be presenting something related to
No. It's Bonnie.
You mentioned for me this book.
Yeah, you mentioned, so now that I remember, we don't store the FASTQ as a GC, we store them as a cube, which is a little bit more compressed. But also, you have the problem that those systems, they are not supported anymore, right? So the algorithm is not supported anymore, so we have to change to something different now. So I don't know what we're going to do next. So coming back to the tools we're using, right? So the idea was we're doing an overview of the tools that you need or what kind of skills you need to learn, right? So one of them is probably sooner or later, maybe your project is small enough that you can use a laptop, but it's quite often that we need to use more power, right? And for that, it's where we use is high performance computing, HPCs, or whatever you want to call them, or even cloud systems like AWS, Google Cloud, etc. So now I have a question for you. So if we need to divide in a so I generally think about how we communicate with computers in two different ways. So can you tell me one? Commands, right? The graphical interfaces, right? Yep. So that's the two main ways, right? So you can use a graphical user interface with point and click, and the computer responds with something. Or command line interfaces, right? So when we think about tools, you can think in the same way that you could divide them in two big groups, the graphical user interfaces and the command lines. And each one of them has advantages, right? I put some examples here on top. For graphical interfaces, I don't know if you heard about Galaxy, but you have other commercial ones, right? Or command lines. You mentioned, a lot of tools that you mentioned when we asked the questions are the command line ones. I don't think anybody mentioned any commercial ones, right? Or graphical user interfaces. Each one has their advantages. One of them is they're easy to use graphical ones, right? You point and click and you get something. They are not that flexible. That's a problem, right? It's complex to create flexibility on a graphical user interface. So with the command lines, you can create pipelines, so they're really flexible. Generally, graphical interfaces are going to be slower. These ones are generally easy to use in high-performance computing systems. As I said, most of them are commercial, so you're going to have to pay. And that's the main issue. Galaxy being the exception. And if I came up with the idea of what they're good for, right? So if you need to do quick analysis, maybe these type of tools are the best. Or if you have a small batch of samples, a graphical user interface can be good. If you have a lot of samples, you're going to probably, sooner or later again, move into high-performance computing and using command lines. There are exceptions to everything that I'm saying, right? So these are general ideas. I especially like Partec Flow. I don't know if you've heard about it. It's a graphical interface that allows you to build workflows, and they are quite flexible. You can put pieces of a big puzzle and then create parameters whatever you want. So at the end of the day, it's pretty similar to what you do in a command line, but in a graphical interface. The main issue is you think, okay, they're easy to use. Well, to some extent, yes, but you need to know what you're doing, right? So in the same way, you need to know what parameters BWA uses, right? In the same way, when you build this type of pipelines and you want to be flexible, you need to be aware of all those parameters and what each one of them do. So Galaxy is another one that I want to mention because we are not going to use it in this as a graphical interface. Can you raise your hand if you heard about Galaxy before or use Galaxy? Oh, not many. Okay. So Galaxy, I think, is a really cool project. So the idea is to build workflows or tools. They have, I think they mentioned they have over 10,000 tools available in their website. So you can do a lot of different things related to NGS analysis. You can build your own pipelines, or you can use whatever other people has built before, right? So you can run analysis in there. Of course, the main issue is currently there are only, as far as I know, 3 public service or 3 public web servers. They have Galaxy. So I think it's US, Europe has one, Australia has another one. It's slow. That's the main issue, right? They're far away. They're generally slow. If you need to upload big number of samples, it's going to take forever. So again, it's the limitation of the graphical interfaces. But if you have a small project, you have one sample, you could start learning to do bioinformatics just using Galaxy. They have tutorials too. So you can go there and say, okay, I want to do variant calling of one sample, and they will teach you using a tutorial how to do it. We're going to do a little bit of Galaxy, just a little bit. Another tool I want to mention is the UCSC Genome Browser. It's a tool, and at the same time, it's a database, right? How many people have used or heard about the browser? A little bit more. Okay. Yeah. But it's still not everybody. So this is the view of the web browser, or one view that you can have. I mean, it can get even more crowded than this. So it doesn't sound like appealing, but the coolest thing is you can visualize the whole genome there. You can have a lot of information in a website, just in a tab of your browser. And it's amazing how responsive it is for how much information you can put on the screen. So yeah, we're going to use it. So let's do one thing. Let's try to do this exercise. So we can go to the website of the Genome browser. It's called genomeucsc.edu. I'll keep this instruction on the screen. If you can follow them, or if you cannot follow, let me know, and I'll do it myself. But the main idea is we're going to select. So one of the first things you're going to see is in the top left corner, I think it is, you have like a menu, and it says genomes, and a drop down, and you can select the human genome, and you can choose the HG38, for example. And if you want me to do it, I can do it live too.
You could click on the link and do it live.
Yeah. So let's see what I mentioned. We're just going to play a little bit with that. First with BRCA1 gene, and then FOXP2 EX47. Okay. So this is the look. Right, I'm going to zoom in a little bit. So if you go to genomes, sorry, it shouldn't have clicked. Come on. Here we go. I said it was responsive, and it doesn't look like it's that responsive anymore. Were you able to follow, more or less? Yes? Okay. Yeah, you're going to get a view like this, right? So here we have chromosome 11. I don't know, you may have something different in your view. I don't know if this was maybe the last thing that I did here. You have a search bar here that you can search for anything, pretty much, of you can think that is being annotated in the human genome, like a gene or a region or things like that, or even other annotations like you can search for ClinVar or dbSNPs or things like that. So if I type, so actually I have my search here. So BRCA1 is going to show me, it's going to go straight to BRCA1 now. So all these things that you have here, they're called tracks, or you're divided in tracks, right? And it's information about the human genome. So generally, the first one you're going to see, and I think that is always there, maybe you can switch it off, I'm not even sure, is the annotation of the region that you're looking at, right? So here we have BRCA1 gene, and this line here is how long is the gene, and the thick lines are the exons. Everything that is thin in the middle is going to be the introns of that gene. And you can hover it of one of the region. It would tell you, okay, you're looking at exon 10 out of 23. It gives you some information about that, right? And it gives you what transcript are you looking at, because each of those lines are different transcript or isoforms that have been annotated for the genome. I think in the exercise I mentioned that actually have it. If you go, sorry, this probably is not on your view, but if you go at the very bottom here, you have all the tracks available. So all this is, if you switch it on and off, you can see it on top of the track. So for example, I mentioned if you go to ClinVar, which is this one here, that is now activated, you're probably going to see this if you go to the ClinVar section. It says hide, and you can put it, let's do dense, for example. This is the way it's going to show that track on top. So dense means is compressed. If you do full, it's going to be very extent of information. I'm going to do dense. I'm going to refresh. So if I go back up, sorry, all this is ClinVar already, but it's full, sorry. You're going to get a new track. Okay, here we go. It's reloading. We still have BRCA1. And here we have ClinVar annotations. ClinVar is a database for variants associated with clinical outcomes. So it can be pathogenic. There's also, so ClinVar is a bit complicated in the sense there still can be, they're classified based on the pathogenicity, right? They classify as being pathogenic, likely pathogenic, benign, or unknown, what they call unknown significance. We don't know exactly if it's pathogenic or not. There's a lot of type of variants, not only clinical related. There's also levels of classification. They give them stars depending on if multiple people have seen that variance being associated with disease. So it's going to have multiple stars telling you, oh, I'm confident that this is a real variant. There's still a lot of false positive, and it's a big effort nowadays to clean up the system. So they're removing those false positives or changing the classification. So of course, BRCA1 is a gene related to cancer is going to have a lot of clinical variants annotated. And if you want to extend it or look at one specific one, you probably can click on one of them, just randomly. It's going to extend it. Yeah. Again, I said it was responsive, but it's not that responsive anymore.
This color indicating
Oh,
all the
I'm not even sure what ClinVar says here, but yeah, it must be if it's pathogenic or benign or things like that. You're probably going to have to check. Pathogenic, right? So red means pathogenic, blue probably means something different, right? Oh, they're all pathogenic? Oh, yeah. So these big lines probably means it's an indel.
There's about the type of variant and the pathogenicity of the variant.
Oh, you see the star system? So if you scroll over one of them, you can see here on top, they give you 1 star, right? So it means it was a single person that submitted this indel that is expanding from that position to that position. So you don't have much confidence that being real if only one person have reported this to ClinVar, right? But if you have more stars, you can be more confident that that variant probably is real. So if you want to try something different, I mentioned to look at this other gene. It's called FOXP2. So it's already searching, yeah.
So this software will tell you the clinically defined variants, right?
Yeah, so the ones that somebody reported to
We
me.
get the information from this. So if we want to check some genes like BRCA1,
Yep.
BRCA2, we will first look into the clinical variant, what this software is giving us.
Okay, so this software, the only thing it does is to show you data. And it can come from a lot of different databases. The only one, what I was trying to do is to show you one track, which is the CleanBar one, but you have a lot of different tracks there, and I'm going to show you a different one now. The CleanBar database is basically a database that is not this, it's separate. So this, what it's doing is taking the data from that database and showing it to you. The CleanBar database is just that, a database of clinical variances related to some clinical outcome, right? So yeah, now we have a new gene, which is the FOXP2. In the same way, it's very long, or it seems long, or there's a lot of big, long introns in it. And these vertical lines, you don't see them, but they are exons of that gene. So I still have, these are the clinical variants again. Probably, I still have it active. As you can see, there are less. Oh, here we have them classified by the interpretation: pathogenic, likely pathogenic, variants of unknown significance, likely benign, and other. So these are the type of classifications. But you can see that this is less crowded than BRCA1, right? And we have less indels reported here. These are just single base annotations. So these are the IDs for dbSNP. I don't know if you're aware. This is a different database on just people report SNPs. So let me go down. I'm going to try something different this time. I'm going to switch off CleanBar, so I don't want to see CleanBar. And let me go. It's still refreshing. So if you are done refreshing, you can try this multiple alignment. Okay, now it's refreshing. So before I activate multiple alignment, another thing that you can show, if you want, you can drag the track, so you can move it left and right, and it's going to move. It's going to refresh and only refresh that window. So you can, navigate the genome really easily on this website. The other thing you can do also, if it stopped refreshing, let me reload. So this is exon probably 3, exon 4.
The transcripts in the UCSC, are they mentioned in ENSOM transcripts? ENSOM.
The ensemble, yeah.
Why is that? Because it is from the US and not from the European consortium. So why do they choose to?
Yeah, I don't know. Maybe you can have both. You can have your own custom track. So if you go to another database, you can get a bed file and upload it here.
So on UCSF, if you want the equivalent, like NM for the same ENST or the ENSOM transcript, can you do that?
Probably you can do that. Yeah, I mean, you have NMs here on the bottom, I can see in this section. This is the annotation of UCSC, but I'm assuming you have all other type of annotations. I don't play enough with UCSC Genome Browser to know everything what is here, but I mean, you can see there's a lot of tracks to use. So one other thing you can do is, for example, I think this is chromosome intron. This must be exon 7. So I'm going to try to zoom in. Sorry, I'll just move it again. Oh, by the way, here on top, you have that red line is the area where zoom in. And of course, you have the zoom in and zoom out option, so you can have a bigger visualizations. Let's see if I can. We said that was exon 7, right? Yeah, that must be it. So I'm going to try to zoom in. What is the exon 7? Is this one? No, don't do it. Let me do it better. Ah. So you see, I just drag and drop, selected an area, and in theory, I'm going to be zooming to exon 7 now. Is this exon 7? Exon 8. Okay. Let me zoom out. Let's see if I can do it manually. If it's going to take too much effort, I'm going to quit. Okay, exon 7, I found it. So this is exon 7. I'm going to zoom into exon 7. And if you keep zooming in, you're going to see the sequence itself, the DNA sequence. We actually see now the protein sequence. Right? In the human, you have the different isoforms or annotations in different databases. So if we go down, and for example, I said, I don't know what I did now. I hope I didn't move it. I went back, right? Yeah. Okay, exon 7 again. So by the way, there are multiple genomes. There's not only the human genome. You can have other genomes. So if I select this, oh, I already have it. Let's do dance. So what track is that one? Oh, here we go. So you can have even your sequence being aligned to other species if you want to, right? And I picked that gene. I was trying to find genes that are interested. I picked this one because this is related to language. And if you can see, pretty much it's a well-conserved gene across the multiple species, right? Except there's only a couple of places that unique to humans only. So there's only one amino acid change that seems to be completely different to the rest of the species. And there's theories about that amino acid change being important for us to be able to speak compared with other species, right? I don't know, I just wanted to show up a little bit the capabilities of UC Genome Browser. It could be a really cool tool for you if you want to explore the genome in a website. Although it looks like an old-fashioned website, you can have a lot of information in a single tap in the browser. So coming back to so another cool thing is that, well, I explained all this. I mean, we don't have to do it. I mean, we already spent enough time. But if you want to, you can look for this SNP. This is the SNP definition or ID in the DBSNP database for one specific SNP seen in humans. And it will show up in the track as being pathogenic, right? We don't have to do this exercise. We're going to skip it. But this type of job you can do. If you have a DBSNP annotation, you want to know if it's significant for something, you can go here and search for it, and you have all type of annotation, what it is in the genome, if it's conserved across different species, if it's clean bar or not, interesting, things like that. Another cool thing about the genome browser is it's connected to Galaxy. So if you want, we can do this exercise, or you can try to do it. Let's see if it works. You want to do it? So if you go to to UCC Genome Browser, oh, this is not the exercise, my bad. So I'll show you in a second. But basically the idea is you can connect whatever you're doing in the genome browser, you can connect it to Galaxy. You can send it to Galaxy for more analysis. So we can do this exercise. So let's imagine we want to get all the coding bases of BRCA1. We want to remove all the introns. We only want the coding bases. So what we can do is we can go to the genome browser. If you go to Tools, Table Browser, and you do this, you're going to end up with what we call a bed file, which is going to be defining all the exons for that gene. So please try it. I'm not going to do anything else. You have the instructions. I think you should be able to follow them.
Yeah, just do it and let us know that we take it. And we have three wonderful volunteers. This is the third wonderful volunteer. Okay.
Or I can go back and forth and show you at the same time and keep the instructions.
If you need any support.
So we have other people.
Yeah. So
any help you need, like, or you can sit there and अपने आसपास के लोगों को बता दो भी आपको.
Once you're done working, guys, just help them.
अपने रूम के आसपास.
So this exercise is only to show you how you can connect two different, or in theory, two complete different websites and use the tools of one with another, right? Any problem finding the table browser? So you have two ways in the UCSC genome browser. You have two ways to look at the genome, right? One is the one that I already showed you, which is navigating through the visual part of it. But you can actually use a table system. They call it a table browser, where you just say, oh, I want all the information related to BRCA1. You type the name, and you're going to get some tables about it. Can I help here, maybe?
So table browser.
Yep.
So yeah, we have to
Yep.
select
So, yeah. So basically, I'm going to tell what you're doing in every step here if you want to. So basically, we're asking, oh, can you give me the gene predictions, the annotation for that gene, right? We're asking, okay, what database I'm taking that information from. So I'm asking for RevGene, that's a database of annotations. This is just telling what region we are looking at. And now we're telling, okay, just give me the information of BRCA1. It's the only gene that I care. And then how you want the information. So I'm asking the output that I want is a bed file, which is this, which basically tells you the chromosome starting and ending position of something. Because we ask about, and at some point did I? Oh yeah, at some point we're going to ask what type of information we want, which is the coding exons. Yep.
If we click send query to Galaxy.
Yeah, so when you're done everything, you can say at some point you get output, you're going to tell you coding exon and you send it to Galaxy, right? The button changes and says send it to Galaxy. And you're going to be sent to the Galaxy website, right? Oh, you need to follow me? Sorry. Yep. So at some point it's going to ask you send the query to Galaxy and you click send it. Just keep it on the screen. That's going to be fine. Okay, yes. So the people who already reached Galaxy, you're going to see on the right side some spinning wheel. So so the Galaxy is trying to retrieve the data that you requested. Yep. So should I do it on the screen now?
Yeah, we are here.
Yeah. So coding exons. Yep. And then you send it to Galaxy. Yep. So I'm going to do it now, right? So I'll show you basically what you see on the screen. So you see, oops. So I went to the genome predictions. Do I need to change anything? No, right? Reference genes, genome. So you've done all this. You selected all the different things. Oh, BRCA1 already did it. So pretty much everybody reached that point, right? So we said the format is going to be a bed file, and we're going to send it to Galaxy, and then you click get the output. I mentioned to select only the coding exons because we only want the coding bases. And you send it to Galaxy. Yeah, we did. I went too quick regarding Galaxy, but this is the Galaxy interface, right? On the right side is you're going to have some folder with your data. So here you can see our data being loaded to Galaxy, right? We need to wait until it gets loaded. And on the left side, you have all the tools available for you. All these are just tools that you can use for your analysis. You have a section for workflows. Those are the pipelines. I don't know where the tutorials are. I don't know if they are also part of the workflows, but I know there are tutorials available in Galaxy that you can follow for your analysis. Okay, so it's been loaded now and it's ready. So I said that we're going to be merging, right? So if I go, oh, well, let's visualize it first. So if I click here, see, I just click to my data, and now I can see the bed file, how it's being created. So that's the bed file of all my coding exons for BRCA1. So, but there's an issue with this. Can anybody point why you want to merge this file and what it means merging? You have an idea why I'm doing this? So the problem when we do this is we're getting all the coding exons for every isoform, right? So there's going to be exons that are duplicated or exons that overlap with each other. And I want only the coding bases. I don't want to have multiple copies of the same exon or regions that overlap. I want a single track that is going to define all the the bases that are coding. So one thing that I could do is we're going to use, so I forgot how it was called. It's called operated. So let's do it differently. Let's go to, here we go. Operate on genomic intervals. And there is one that I should call merge. And already detect that I have some data here. And it tells you what it does. There's an explanation. And this is what we want to do, right? So our data set contains coding exons that overlap with each other or they're duplicated. And I'm only interested about the coding bases. So if I do that, I can run the tool. Now you have a different set. So it's thinking. Now it's working with it. And in a few seconds, I'm going to get what I want, which is a bed file with all the coding bases. So this is the type of things you can do connecting the UCSC genome browser with Galaxy. I mean, Galaxy is more than that, right? Galaxy, you can do it, as I said. Everything we do, by the way, everything we're going to do here in the workshop, you could do it in Galaxy. You may do it slower, maybe, depending on how many samples you have. But everything we're going to do in this workshop, more or less, except plotting in R, everything, all the analysis we're going to do here, you could do it in Galaxy. Except the single cell, maybe. I don't know how the tools in single cell are here, but everything else, you could do it in Galaxy. So it just finished. So now we have a set of unique regions that define every coding base. Now, you can use this for variant calling. You can use it for different things.
So it is giving information about all the transcripts of this gene.
So originally, when we went to the genome table browser and we asked, give me all the coding exons, it gave us all the coding exons for every isoform in there, right? So, and what happened? And then we decided, I only want coding bases. I want a unique set. I don't want to have multiple. So if you compare this file with the original one, you're going to see that a lot of sections overlap or they're duplicated. And I want a unique set of coding bases. So that would be a way. You just did the merging operation, right? Which, sorry, I went really fast to this. Sorry, come on. It's giving you every base on that gene that has been annotated as coding. So this was our situation, the red one, right? So our original set, it was defining regions that overlap or were duplicated. And now what we get is the merge intervals, which is the bottom one. So the first we have red, and now we have blue. That's the main idea. But it just was a test. So I wanted to show how these two data, so these tools that I think is going to be useful for you guys, you can connect them, right? We are not going to use these tools anymore, by the way. Sorry. It was just a demonstration of graphical user interfaces. I wanted to do something with a graphical user interface. From now on, it's going to be more manual work. Coding, right? So as I said, this is something that sooner or later, as a bioinformatician, you're going to have to learn. I'm not going to call it only coding. It's programming skills in general. It could be just command line, know how to use the command line interface. It could be actually coding code. But sooner or later, you're gonna have to, I think, right? So talking about coding. So I would say nowadays R and Python are the two most popular coding languages, scripting languages. So scripting meaning you don't have to compile the code to create a program. You just type your commands and you can run them. And there's an interpreter who's going to understand what you're typing and doing something with it. So R and Python are the most popular ones nowadays. So actually, I'm not trained with R. I'm not trained with Python either. I used to be trained with Perl, which nobody nowadays uses pretty much, right? Everybody has moved to Python and R. So even to me, it's hard nowadays to code myself or to talk about myself as somebody that programs because I'm using skills. I'm losing skills, I would say, right? I spent I don't know how many years, 12, 15 years programming in Perl, and nowadays it's like, okay, we move on with Python and R, sure. It doesn't matter what you learn, right? Maybe in 10 years from now, we quit using Python and we start using more modern languages. But anything you learn now, so a lot of these languages have commonalities, and you're going to learn structures, how to structure your program, and everything else. So you're not wasting your time if you learn a language now. Although it may change 10 years from now what we are actually coding in bioinformatics. I make also an effort to try to define how different they are from each other. Nowadays, there's a lot of overlap too. You generally say, oh, R is really good for plotting, right? Creating graphs and all that. It's true. It's maybe easier for that and maybe more difficult to learn. For plotting, maybe easier, so that's a good thing. But any library that you have here, it's moved to Python too. You have matplotlib. It's a library that allows you to plot really cool things, similar way that in Python does. Although it's more, I think it's a bit more complicated. And the other way around is true. Some of the libraries that used to be only in Python are available in R, or you can connect them. You can incorporate Python code inside of the R code too. But yeah, the general idea is if you try to say, oh, I need to decide what are the two languages I want to learn, you need to think about what type of data you're going to be analyzing, if you want to do a specific plot or not. Nowadays, I would say single cell is there's a lot of application that we do with single cell, and I would say R probably is the most popular for that. So if you're working with single cell, then probably use to R. But again, tools that exist in one also exist in the other. There's a lot of overlap. No, you're not going to be wrong choosing one or the other. If you have to choose, of course. You can learn both or use both. There are other type of tools that are popular in bioinformatics nowadays. Have any of you heard about these things? Conda, Nextflow, or Docker? Well, some people, okay. So this is what we call package managers. You probably, if you work with Python, you use pip before. If you need to install a package, you pip, put the name of the package, and pip take care of anything you need to know about that package and installs all the dependencies and everything. So that's good. There's also the equivalent in R. Conda, what it does is a bit different in the sense that it's also a package manager. If you need to install a software, Conda is really cool because it doesn't matter if it's a Python package or R package or a command line tool or anything, Conda takes care of it. The problem when you install a software, if you're used to the graphical interfaces, is you download the full package and everything is there. Nowadays, there's a lot of people coding, and one software depends on another, another depends on another, another depends on a third one, and a fourth, and so on. And it's really hard to have, okay, I need to install this, but before I need to install that, because that needs that needs that. So it gets complicated. So this type of tool helps you with that. I'll talk a little bit more about Conda in a bit later, but Conda does things a little bit different. Nextflow is becoming really popular. Again, probably everything we do here, you can do it using Nextflow pipelines. So Nextflow is a workflow manager. So if you go to the website, you're going to see there's a lot of pipelines there. So if you need to do variant calling, you can go to there, look for a variant calling pipeline, download it, and you have everything you need to run your analysis in a single package in theory, right? Nextflow can use Docker and Conda inside of Nextflow too, or it can be combined, all those things. Docker is a bit different, but related to the other ones too, in the sense that it's the Nextflow is clearly for bioinformatics. Docker is used for everything. So let's imagine you need a specific software, but that software, again, needs dependencies that you need to install several other things to make it work. You can get a container, they call it. You can download a container with everything you need in that single container and run it. It doesn't matter if you work in a Mac computer, you can work in a different environment. It should be supposed to have everything you need to run whatever you need. So you need to be a little bit aware of these things because it may save you a lot of time when you need to work in bioinformatics. Don't worry, it still doesn't work, so we're not going to do it again. But this is something that we wanted to think about it, right? So do you use ChatGPT? Yes? Can I see, how many people use it every day? Just to have an idea. Oh, wow, so everybody. Bioinformatics is not different. It is not. So you can use it pretty much every day, right? So how we use it, I was thinking, okay, how I use ChatGPT every day. So for writing code, you can use it for writing code. I'm not talking about tools specialized for something, or the language models, you have the different types of large language models, LLMs, that is being specialized to PubMed searches or reading papers for you and all that. No, I'm talking the ChatGPT generally, right? You can use ChatGPT for writing and debugging scripts. So if you have doubts about one piece of code, you can paste it in. ChatGPT will try to correct it and so on, right? It can even explain you the code, what it does. It does a pretty good job doing these things. It can annotate code. I think this is really useful sometimes. If I have code, I never annotate that code. I go there and say, please annotate it for me. And it's going to explain every line what it does, so I don't have to do it myself and type it. I don't know if you try this. I never tried this, but it probably can do it, right? You can probably giving, oh, I obtained these results, what it means. I'm assuming ChatGPT is going to give you an answer. Always gives an answer, right? But that's a problem too, which is there's hallucinations. I remember when ChatGPT came out, I was trying to use it to get papers, right? I remember asking, okay, give me the top 10 papers in that field. Give me, because I want to just have an idea, and give me the PubMed ID of those top 10 papers. All the PubMed ID were wrong. Not a single one was correct. But ChatGPT was convinced, here you have the 10 papers, here's the PubMed ID. The titles were correct as far as I remember, but the PubMed IDs all were wrong. Data privacy is another problem nowadays, right? I mean, Yale is now pushing a lot to have rules. We have different LLMs in Yale that are they have some privacy settings on them. There's one that's completely private. There's others that take your data, and they don't want us to use ChatGPT openly, right? I don't even know which one. Maybe Bonnie knows the ones we use. I don't even remember. Of course, there's no way to reproduce your data, right? If you use ChatGPT for an analysis, I don't know if you're going to do that, but it's going to be hard. In a month from now, it can be a completely different ChatGPT from the one that you used 4 weeks before. And they can have biases, of course, depending on how the data was trained. So you're already aware. I don't have to give you a class about this, you know. And of course, machine learning and all that would need a completely different workshop. Oh, and this, right? So you need to remember this. Always say thank you, because 10 years from now, who knows what's going to happen with the AI. So, but what tools are we going to use for this workshop? So our cluster, our high-performance cluster is going to be AWS. I don't know if you're aware of. So basically, Amazon, if I'm wrong, if it's true or not, that's what I remember long ago. I mean, I remember using also in a workshop like 15 years ago with AWS. It changed a lot completely from that time now. But I think Amazon, what would happen is Amazon sells stuff through the year. It needs some computational power for whatever sales it does. Christmas comes, all of a sudden it needs 100 times more computational power to be able to sell everything they want to sell. They realized that through the year, 11 months, their computer power was idle. It was not used for anything. So they decided, okay, we can create a business out of it, which is why we don't sell the computational power to the people, and they can use it. So that's how AWS was born. That's as far as I understand how it was born. So yes, so that's going to be our HPC is going to be AWS. you don't care much because you're not going to see it that much, right? Your laptop and a browser is going to be the windows that are going to be used to connect to the AWS. Conda environments, that's what we use to install all software in AWS. We use a Conda. So we create a Conda environments with all the programs we use to the workshop. That is not something that I recommend, and I'll tell you why, but you can do that. We're going to use the Jupyter Labs and notebooks, which is basically, if you use RStudio, it's similar to that, but I think it's cooler. That's what I think. Inside the Jupyter Labs, we have access to terminal, so we can use the terminal if we want to, but actually you can do everything through Jupyter notebooks if you want to. And some people mentioned about RStudio. You don't need to install it or do anything with RStudio. We're going to use the Jupyter notebooks, but if you want to and you want to use it, there's ways you can do that. So if you want to install RStudio and use RStudio on your laptop, there are times you can do that. So yeah, so these are the tools we're going to be using. And I want to mention something about Conda, right? The advantage of using Conda when you need to install something, you can, by the way, you can use Conda in Windows too, or your Macs or any kind of Unix. You can use Conda. Conda has an advantage, and which is, first of all, allows you to create different environments, we call it. So let's say when you install a program in Windows, it's immediately available to you, and it's always available to you. When we work in bioinformatics, there's issues with that, which is programs change so fast that you need to update it every month. Let's imagine we work with CRAD, which is a tool for single-cell RNA-seq analysis. I do my analysis today. 3 months from now, there's going to be a new version. And I said, OK, let's install the new version. It's going to override the old one. And now all of a sudden, I need to reproduce the analysis that I did 3 months ago. I cannot do it because the version is different. Right? So Conda helps with this, for example. Right? So with the Conda creates what we call an environment. It was a space of programs that you install. You can activate it or deactivate it anytime you want. So let's say I install everything that I need for analysis today. I create that environment. 3 months from now, I want to update something. I'm not going to update it. I'm going to create a new environment with the new packages and create that new environment. If I need to go back, I'm going to say deactivate the new environment, activate the old one. And now I have all of a sudden the exact environment that I use for my analysis. The cool thing is I can say, oh, my friend wants to repeat exactly what I did with exactly the same programs that I use. So I can send him my environment. And he can reproduce the analysis created in that environment. So the way to encapsulate is really important for what we do. And I think Conda does a really good job. Before I said I install every single package for this workshop using a single environment, I don't recommend it because you have a lot of issues because there's incompatibilities with some programs that we use. So generally what you're going to do is if you work with single cell, you're going to have an environment for single cell. If you work for variant calling, you may have a different environment for variant calling. And within those, you may have different versions of the packages. So if you look at the one that I use at Yale, I have, for Seurat, I think I have 4 to 5, depending on the version that I'm working with. And that happens. So you may end up having 10, 15 environments. But that's fine. I mean, not all of them are active. You only activate them when you need them. So, and one thing that I need to mention, so in Conda is that part here. This is important, right? I didn't use the word reproducibility, but we need to use it. So we are constantly being tested about reproducibility in science, right? Not only in the lab, also in bioinformatics. We need to be able to express what exactly we did so somebody else can reproduce our studies. So Conda will help you with that. Somebody can ask you, well, what software and versions you use? All of this. Here you have the list. Conda is going to give you all the versions and all the packages you use. Jupyter is similar for that, right? In the sense, it helps you a lot with reproducible science. So Jupyter Notebooks, what it does is combines code and annotations in a single document. So you can start a document, it can be text. All of a sudden you want to create a plot. Let me create a plot in R. Let me document the plot. Oh, I need to run some code and something different. I run a code. So you can document the code in a single document. And it's also helpful in the sense, OK, what did you do? And how you do it? Can you explain me? No, I don't have to. I give you my Jupyter Notebook. You just read it, and you do it. So documenting and sharing, another important part in bioinformatics that we need to learn to do, right? Or, I mean, 10 years ago, it was that I share my code, but it's not even annotated. It's going to be hard to read. So this type of tools helps you a lot. So Jupyter Notebooks are really useful. RStudio has its own way to do notebooks. I like a little bit better in Jupyter. So that's why I think it's cooler. But RStudio also works. Originally, it was designed for Julia, Python, and R. That's where the Jupyter name comes from. Nowadays, I don't know, you can have a lot of different languages. I don't even know, but there's a lot of them. There's even ways to mix languages in a single document. Actually, we could mix two languages in some of the documents, but there's ways to even mix R and Python code in a single document, and command lines too. And I think it's 1, right? So it's a good time to stop and have lunch. Yeah? Okay, so we're going to stop here, and when we come back, we're going to start working with Jupyter Notebooks.
For the 5 p.m. talk, we're going to try to connect this.
Yeah, mic is working, right? Yeah. Yeah, you see a website like this? Some window like tells your username and all that. Okay. So I want you. So the username for each one of you is going to be the email that you use to register, right? So if you register with that email, you made a mistake because that's my email first. But second, if you register with your email, what username is going to be is the whatever is in front of the domain without the dot. So my username is going to be this one here if I register with that email. So you remember the email that you used for registering into the workshop? So your username is this, the part in front of it without any dots. So try. Password, now you're going to choose a password. It's going to be the password for the rest of the workshop. So try to use something that you remember. It's going to be your personal password. Yeah, you can create the password now. That's going to be your password. Oh yeah, and please remember the password because it's going to be the same one for tomorrow, the day after, and so whatever password you choose now is your password for the whole workshop. Are you guys able to log in? And you see something like this? Okay, good. So,
Please don't enter your domain name, just your the
the,
first half of the email, right?
and without dots. So if there's a dot or dash or something, don't use it. Oh, so it says everything here, right? No domains, no dots, lowercase. Oh, yeah, totally, yes, yeah. Whatever is your email, yes. I'm gonna move.
So please remember your email address. What is your email address? So put type it down. That's it. Password. That's it.
So everybody's in. Well, let me ask you differently. Is anyone has any problems with it or logging in? Yeah, everybody's in. You have a problem. Don't move it.
Right? So please mute that one topic, right? Some of them are not registered, so that is why.
Oh, yeah. If I don't have your username, I'm not able to create it. Yeah.
Lab participants, announcement for the lab people who are in the lab. There are groups for 2 people, so only one username will be, given. So log in from one system only. You need not to log in from 2 different systems, please. So like me and Kajol
That's a good point. I don't know. I never tried. Maybe it works. Yeah. No, I don't think so. Oh well, yes, they can mask if they cannot modify the same file. That's the problem. Yeah. Yeah, modifying the file, no, but looking at it, yes, kind of thing. They can probably modify different files, which is fine too.
Yes, but at the same time, not on the same file. Because
Not at the same file, no. But for now, I think today should be fine.
yeah, because they're not doing anything today.
Okay. They're gonna do things, but they could duplicate the file and each one to have one. I guess that would work. I guess.
It'll be a derivative of the same. हो गया login.
Um, so we're missing 10 people. Could be that it. So there are 10 people missing today?
How many?
10. There are 10 people that sign up, but they are not here today? Oh, okay. So I have this, I can see that there are 10 people that don't have login yet. So I don't know if everybody.
No, they are sitting here. Let me just see their names.
So that.
Ravi. Ravi, where's Ravi? Yeah, he's here. No, Ravi, Pratibha, Vishali. Oh, so these people. Sanya ka nahi hua aise login. They're trying to log in. Some of them. Okay, Khemraj is not here.
Oh, okay. You're still doing things. Okay, sure. Yeah. I was just checking that to be sure that everybody's.
There will be like 4 or 5 people missing. Otherwise, I think it is good. Can you name them who are missing? Like for.
I don't dare to pronounce all the names, so.
I'll do it. So Neha Jindal.
Oh, I can do that.
No. Sudhanshi. She's not here? So she's a participant who's missing. Yes. They paid for it. So Ravi Pratap. You've logged in, right? But
Are you in?
are you in? You've not done it? No, but you're there, right? This is open.
Oh, okay.
Okay. Maybe.
Oh, I have less now.
Yeah.
Sorry. There's 7.
So, no, but Ravi is still not logged in. You're still not logged in.
Hey, but this system will log in.
Japneet. You're logged in. So she's also logged in. Vishali is not. She's gone downstairs. Khemraj is taking
Okay.
class. Two.
Okay.
So 2 are missing, but rest are here. So Vishali and Khemraj are not here. I know they've gone downstairs.
So okay, let's see how we do. Let me do one thing. So you said you are login. You're login. So is your username here? Is any of this?
This one, Ravi Pratap Dope Sagar.
And that's the one you use for login?
Yes, sir.
Is the same? Maybe it's a. Because if it's not the same, then he can't log in.
Okay. Yeah, I know. But that's surprising though. Let's see.
कितने लोग लॉग इन नहीं है?
Sir, इसके हिसाब से तो 8 नहीं है, but they are Pratibha and Ravi. They have logged in, but it's not
नहीं वो
reflecting.
जर्नी भी
हाँ, Japneet also. Vishali and Khemraj will log in नहीं है, they're not here.
So it seems that the system. Yeah, no, I can see that for whatever reason the system added something extra to their login. You didn't log in like this, right? This dash FD whatever. This is not you, right?
No.
Okay. So the system itself changed the name. I don't know why. So I'm gonna modify it. Oh, I know what I'm going to do. Okay. So now that, let's see, now that everybody is in, you're not going to have exactly what I have on the screen. It's going to be similar. We'll fix it. I see that some of the usernames, the system changed them. I don't know why. It added extra letters that you don't need to add, but that is why I don't see them. You're going to have a view like this without this section here, right? Okay, forget about this for now. We'll figure out how to fix that later. Okay, so this is what we call the Jupyter Labs, right? So you have this view here, which is you are in your browser. So this is your browser, and you have this tab with all this information. So this section here, they call it the launcher. Here is where you're going to start. You have different applications. We call it applications, I guess. You can start notebooks, R or Python currently. That's the only thing you see. You can even interact with the console, the R console or the Python console. I don't know if you know what it is, but basically is your RStudio without the graphical part of it. Yeah. There are other applications like you can use the terminal, so we can use command lines and directly interact with the terminal. You can do it. Just, let's do an example. Start the terminal. Click on the terminal. And what is going to happen is this is going to transform to the actual terminal, right? So if I click, right? Now you have something like this, a prompt. We call it the prompt, right? So you're going to have your username here and some number here. Forget about that, but that's the prompt. And you can start writing commands and doing things, right? If you do an ls, for example, that's a simple command, right? If you do ls for listing, it's going to tell you that it's empty because you don't have what I have, but you will have something. So if you want to close it, you just click on here, and it will go back to the launcher.
I think so it's not the same as what you see here, right?
Yes, don't worry about this section here. But can you see the rest? Yes. Because you don't have it. Yeah. That's what I'm saying. Yes. Don't worry about it. We're testing now. You can play with it. You're not going to break it. Okay. Don't be afraid of testing things because you're not going to break it. You're not going to have the same as I have for now. Don't worry about it. The what? Don't move it.
When I type ls on the terminal,
Yes.
it didn't show me this.
He show you something? Oh, yeah, because you don't have anything yet. We need to solve the issue with some people that have different usernames. So when we solve that, everybody will have the same as I do. So for now, forget about this. You don't need it. Okay, so this is a launcher. And you can have as many launchers as you want. You can keep opening tabs, right? And have different things. So this is a tab inside of a tab, right? It's a little bit of Inception, the movie. So so you can have as many as you want with different applications. I'm just showing a little bit what you see on the screen and you're going to see. Another important thing you're going to notice is on the left side. So to go back where? Close it. Or close the tab. You see the X on the... Yeah. Don't be afraid of try. If you don't see something, try. And if you try 2 times and you don't know how to do it, we'll figure it out. Yeah, so this is basically where we're going to have our notebooks and terminals running and all that. This is the main window. But on the left side, you also have what we call, it's a browser. Probably I don't have to tell you what it is, right? You probably identified, like you see folders. You don't see anything. You will see in the future because your browser is empty. Your home directory is empty. But in this case, we can see folders, and you can navigate with any browser. This is just like the regular browser on your computer. You have it here. You don't see your computer, though, right? As you notice, we log in AWS. So it's a computer that is far away. Well, it's not far away. It's in Mumbai, yes, in Mumbai. So AWS in India is in Mumbai. So you are connected to your computer in Mumbai. So whatever it is in that disk space is what you would see here. And soon you're going to be, you will see this. But for now, forget it. What else we have? We have a menu with, like if it was the software that you have in your computer, where you can open and close notebooks. Basically, all the functions that you could do down here, you can also have them up there. That's the most important thing you need to know so far, and we're going to be learning more things as we go through it. Yeah, for now, the browser is going to be important for you because we're going to opening notebooks and closing notebooks and things like that. So one thing we can do now is when you don't click onto this R here, which is a notebook for R, just click on it. Yep, you see something like this? Okay, there are two terms that I'm going to be using every now and then. I do want you to learn them, right? Let's call it technical terms. They are not that technical. This is a square thing here, this thing here, and you have some prompt signal here. That's a cell. We call it a cell, right? The other thing you need to know, you see these R here on top on the right section that says R. This is the kernel. So this is a cell and this is a kernel. The kernel is basically, if you try to type a command here, the kernel is expecting some type of command. So this kernel R is expecting you're going to type R code. But you can have a Python kernel, you can have a Julia kernel, and as I mentioned before, I think there are kernels that combine languages. The only thing you need to know so far is we're going to be using only R. So if you don't see an R here, you probably open a notebook that wasn't expecting, right? Or we are not going to use. We are only going to use our kernel, so we're going to use our Jupyter notebooks, right? Oh, my bad. No, that's not true. We may use Python ones too. That's the thing you need to know. So let's do a test. Type anything here, whatever you want. A word. The cricket player that won the GOAT that we decided is the greatest of all time. By the way, who's going to tell him? You're going to contact him to tell him that he's the GOAT? Or he knows already? He knows? Okay. Well, tell him that only 30% of this class believes is the greatest of all time. Yeah, I mean, you can type whatever you want, right? So if I say hi, right? If you press enter, it just creates a new line, and you can type hi again, and you can type whatever you want, right? You can keep typing. Yes? So nothing happens. I'm just typing text. Let's imagine this is our code, which is not, but let's imagine this is our code. So to execute the code of a cell, what you need to do is to press play. You see the play there? So you can press it. Try. Well, it tells you that the R kernel in this case is telling you, oh, I don't know what you're talking about. I don't know. Hi is not a function. It's not an instruction for me. I don't know what it is. Why? Because this is a code cell, right? Another thing you notice, you see that number over there? I executed one time. So this is keeping track of the cells that are being executed. Oh, did you get, I don't know if when you press play, you do get a second cell below it? Yes? Okay, let's try again. So if I say hi again, I'm going to show you a shortcut that you're going to be using a lot. Shortcuts in the keyboard. So if you do shift and press return at the same time, it executes the cell automatically. I don't have to press play. So there's a lot of shortcuts here in Jupyter Notebooks, and a lot of configurations and a lot of settings. Again, the only thing I want to remember now, probably the only shortcut that is useful for you now is shift enter. So where you're going to type a code, shift enter, executes, and creates a cell just below it. Questions so far? Easy, right? Yes? Yes? Okay, good. I've told you that Jupyter Notebooks allows you to write text. Why is not accepting my text, right? Because it's thinking this is a code cell. You see here that it says code? Yeah, so if I go back to the first cell, right, I activate that cell, I'm working here. I could change and say, no, this is not code. This is actually markdown. You see the number disappears on the left, and now if I execute it, I get text. So it knows that it's text. So there's going to be two types of cells that we're going to be working on. The ones that is marked down, and I'll explain what it is in a second, and the one that is code. I don't know, anybody in this room ever wrote a Wikipedia article? Or modified a Wikipedia article? No? Okay, so in Wikipedia, they use markdown language to write the text. It's super simple, right? But it saves time. Instead of giving nice fonts and changing a title and all that, they use what is called markdown. So Jupyter Notebooks uses markdown. If you don't care about the language, that's fine. Markdown language, you can just write text, and it's going to look like this. That's totally fine. Right? But Markdown has a specific way of writing text. So if I double-click again on the text, it shows me this again. But let's imagine that the hi actually was a title. So to indicate a title, I'm just going to use an example. I'm going to type this. And now, sorry. Like this. Now I have a different font that represents a title. I only put a symbol in front of it, right? That's the only thing I did. That's Markdown language. It's that simple as that. During the exercises we do, I'm gonna give you a Jupyter notebook already finished, and you're gonna see titles and all that. But if you double click on the cell, you're gonna see actually how it looks like. So,
Francis, so why you actually use Markdown? So codes are there, but why do you need to write Markdown? For two reasons. One, you need to give as much information as possible in your code, so that if you are seeing this code after 6 months, you should at least know that what you are doing. That's the first thing. Second thing, if you left your lab and somebody else is taking over that project, they also should know that what you did. So being explicit, write everything you want to actually write about it. You can put as much information as possible. A good coder, like a person who is a good programmer, he has to be explicit, he has to be detailed about, like you will write one line of code, but you should be writing a paragraph of information on the markdown, so that somebody should be able to reproduce it, or even you should be able to reproduce it somewhat. A lot of times we don't know which genome build was used, what was the database, and you will be just scratching your head that what I did, why I did that. So markdown is very, very important for that way. So
they know that already.
I don't know.
Yeah, we show it in a slide before we start this, before lunch.
That is the most important.
So let's do one thing now. So we already learned how to execute cells, right? There's no questions about that. You have two types of cells. It's code or markdown. That's the only two types of cells we're going to be using. So if you want to learn a little bit more markdown, we're going to do an exercise now. So if you go... Raw is text. Just raw text. Yeah. If you go to help and you go to markdown reference, you're going to see something like this. You remember I try the title one, right? The heading one, right? I put that symbol. Markdown execute, when you execute the cell, understands that he needs to change the font to this, right? I can put subtitles and it's going to be heading 2. So there is, if you need to learn how to use it, you can go here and use it. But let's do one thing. You see out here you have the 10 minutes markdown tutorial. Let's do it. Click on that so you can practice markdown. And we're going to use it on one of the notebooks that I have next.
Begin lesson?
Yeah, begin lesson, I'm practicing. It's going to tell you, create a title, and you're going to create a title, and so on. It's self-explanatory. You don't need me for this. Yes?
So where do we have to sell it?
So once... So people is asking me how we got to the tutorial thing. So if you're clicking help, at the very bottom, I think, or one of the last options, it says markdown reference. Click on that, and you will see the tutorial part of it. So when you started a Jupyter notebook, a file was created, right? So now in your browser, you have that file. Yep. So you can remove it if you want to. If you don't like it, or you could do right click and delete. You could delete that file if you want to. If you like it to conserve it and you say untitled is not an explanatory name, you can rename it too. So I don't think I need to explain how a browser works, right? Everybody knows. Okay, again, to access, you go to help, markdown reference, and you can start the lessons. Yep. Okay. Repeat. Yeah, sure. Okay, who needs me to explain it again? So I'm going to do it again. Nobody. So everybody's good? I'm going to set it again. So if you want to do the tutorial on how to use Markdown language, you click on Help. At the bottom it says Markdown reference. You click Markdown reference and just say, try our 10-minute Markdown tutorial. Click on it. And you can start beginning the lessons. Now you have the files, right?
Yeah.
Okay, good. Do you mind? Oh no, keep working. Keep working.
He's doing something else. We are the one.
You have an account. You should do it.
Is that good?
Yeah. 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 You got to prove. You have my approval. I don't know how it's useful for anything though. I just asked for it. Right, it's a mic on. Sorry. Somebody must have one of these, right? somebody must have one of these here. You can use somebody else's. Oh, we have a, okay. Thank you. Did anybody finish? Did everybody at least reach 4 or 5 lesson? 4 or 5?
Yes.
More or less? Yeah? Okay, good. We can stop here. We don't have to do the whole thing. Um, we're going to go straight to practicing. You're going to learn more practicing than just the tutorial stuff. I think you'll learn the basics. You understand how the Markdown language works now, right? A little bit? Yep. So, yeah, everybody close the tutorial. So you just need to close the tab. And you go back to the launcher. Does everybody have this in the browser now?
Yes.
Everybody?
Yes.
Okay, good. So I guess everybody's now here. So in here is where we're going to have the materials for every day. So every folder is going to have their own notebooks and everything. So we are in day 1. So I want you to open that folder. Right? Use the browser and open the folder called day 1. So you're going to see one lesson that says 00 lesson I, pi, and b. Yeah, so when you see that symbol, that I don't know what it looks like the notebook symbol and the extension I, P, Y, and B. So that is a notebook. So the extension is Interactive Python Notebook. They use it now for everything, but it can be R, it can be other languages. But that's the extension of a notebook. So double click on it, like you're opening a file. Or that probably you cannot read it, right? Well, you don't have to read it on my screen. You probably can read it in your own. So I'm going to remove the browser from the middle, right? So now it looks like a mess. You just see just random text on the screen. Do whatever that every cell says. try to go through that notebook. The first cell says add markdown cell above this one, right? Add a level 1 title that says this. By the way, this is an actual paper title. So how we add a title above one title? So you see here on the right, it says plus below and plus above. Click plus above and you would add a cell above that one. Yep. Just, yeah, scroll over it and you will see it. So you can even double click and you're going to see what I did. But what I want you to do is to go here. If you click at the cell, add the cell above it. I know this is below. Sorry, my bad. Oh. Now.
So when we click that folder, the side, the screen is coming like this.
So yes, so if you want to remove the browser out of the way, you click the folder and it hides. You don't have to, you have enough space, but here it's a bit crowded on the screen if I keep that open. Yeah, what did everybody was able to do? Were you able to add a cell on top of it? Basically, I'm asking this, right? So I'm going to copy this, right? Now we have a title for the document. You want me to do the first part again? Okay, and let me know where the problem is, right? So I'm going to remove this. So when you open the document, this is what you see. I'm asking you to add a cell on top of it. Do you see the symbols on the right? Okay, press escape. Let me see. Oh, that's true. Some people don't see them. Huh.
That's right. The major problem is we.
Well, so if you don't see the symbols on the right, if you go to edit, it's edit. Yeah, that's pasting though. Okay, I'm going to show it in a different way. Some people doesn't have the symbols on the right, I'm not sure why. So you could, if you press the plus symbol, it's going to add a cell below. Yep. So, again, you don't have even the arrows, right? That's annoying. Oh, now you have them. So move that up.
When we click on this plus symbol,
Yep.
we got this.
So move up.
Okay, sure.
No, move that one up. Now you have it on top. You need a space. Yeah. And now you can execute it. Yep. Yeah. Keep going the document down. Do the next line, whatever the next cell says to do. You can keep working on the document. So, did we get everybody that got the title on top of the document? Keep working on the document. Go to the next cell and do whatever it says.
फोर्थ को मारना पड़ेगा तो मारना नहीं किया उसके बाद
Did you figure out how to add a level 2 title, the introduction?
Yes, we have done.
Okay. Keep moving. Can you create a table now?
How to create table? Looks like this. How to create table in this?
You wanna know how? Sorry, keep doing. Double click. Skip the step of the table because if you didn't reach that point in the tutorial, you're not going to know how to create tables.
And in the level 3 title we have to create, right?
Yeah, the level 3 title created. Skip the step of the table if you don't know how to create tables. I think not everybody create tables in the tutorial. Tutorial tells you how to do that. You don't have to do it. Don't worry. If you have time later on, you can learn how to create tables. Oh, I know when. Is your document looking like this a little bit? Yes? Did you figure out how to attach an image? Have you tried to attach an image to the document? No? Just follow those steps. So we add a markdown cell below it. We're going to open. Copy the image. Did you try? It worked? Okay, good.
So it's mine is also coming same.
Yeah, so add a mark. This is annoying. everybody reads the formula. I'm not gonna, you don't have to learn how to incorporate formulas, but you could, right? So if you double click the cell, you're gonna see how I did it. Right? It's quite complicated to get the right language, but if you want, you can put nice mathematical formulas in your documents too. if you finish the tutorial, I teach you how to do links on documents, or hyperlinks, or whatever you want to call them. So this part of the exercise tells you, or add a cell below it. It says that this text needs to link to whatever link is that, right? So the way to do it, you need to put it between brackets. That's how Markdown does the links. Square brackets, open round brackets. Oh, sorry. Well, let's do it like this. Copy. So square brackets is for the text that's going to be visible. Round brackets is for the link. So if I execute this, oh, sorry, I didn't change. So it needs to be marked down now. So now you have the link. So now this text is a hyperlink for that. So the only thing I did was this. So square brackets for the text, round brackets for the actual link. Sorry, what? Oh, it doesn't work, right? I know. Don't worry about it. Yeah, saving the document as PDF is not working. If you want, guys, I can try to fix it tonight, and I'll try to fix it, but you don't have to worry about saving as a PDF. Any document that we create here, if you want it, we'll share it. So you don't have to download it. Did you guys execute the code? You did, right? And it worked. Okay.
Stimulated local temperature train तो आ गया, but this figure has
Two things that I want you to learn from this. Well, the shift enter is your friend from now on. If you want to execute a cell, shift enter, shift enter. You need, if something doesn't work, double check that you're actually in a cell that is code or a markdown. So you need to distinguish between the two. That's another thing. So people were asking me how you create tables. This is type of format. You don't have to create tables. I mean, if you do the tutorial again, at some point the tutorial teaches you how to do tables. What other questions we have? This is actually LaTeX. This is how you create formulas in LaTeX. Markdown understands LaTeX too, which is another language to create documents. So if you need to create some complex formula for whatever reason, you can do that. I don't think, no, we're not going to ever create formulas here, but you know that is a possibility. I guess everybody got that graph. So another question that we get is why we use Markdown or the title. The most important thing I want to learn is how to create titles, because the Jupyter Notebook automatically creates an index. Right? Here on the left now I have an index. I know it's quite silly index because it's small, but if you have a document of 10 or 20 pages, you don't want to scroll down an app all the time. And if you've been creating titles and I want to go to the method sections, I go to the index, I click on the methods, and it goes directly to the method sections. So I know what it is. So that is why it's useful to know how to create titles, because automatically you create an index. Okay, we're done with this, so you can close it. And I'm going to ask you to go to the browser and open the other document. You have two documents for day one. We're done with one. I'm going to open the second one. So, yep, we opened the second document. Everybody's here. You have a very long cell here. You don't have to worry. We already done this. But if you are curious of how to do all these things, like how to paste an image and everything, it explains you again. So if you double click, you're going to see how the cell looks like, right? So I don't know, I think you practice how to create bold text, italics, how to cross text. All that was part of the tutorial. How to create hyperlinks. So if you're curious how the text looks like, you can double click anytime, any cell. So if I want to see the format again, I just do Shift Enter, and here it is. We already learned how to paste images. You can even paste GIFs, right? So this is a GIF. So it's not me moving anything. It just pasted the GIF here, and it tells you that if you want to change from different types of cells, you just go here and change from code to Markdown, and that's the way you change what is inside of a cell. It's a cool document, right? You can paste the GIF there. So it's hard to find a document you can do that. Not even Word does it, right? Can you? Okay. So let's start coding a little bit, or at least practicing coding R. Now, I want everybody at this point now. Everybody open the second document? Yes. So if you scroll down a little bit, you're going to see a brief intro to R. Yeah, everybody knows what R is because we didn't even explain it, right? It's a statistical language that is really useful for plotting and doing statistics. What we said before, right? The kernel. That's really important to know to be sure that you are working with the right kernel, right? When you open the document, automatically knows that it's an R kernel because I create it as being an R Jupyter Notebook. So you probably know what it is. This is code, right? Because you have the square brackets here. And if you go here and do Shift Enter, it's going to execute it. Do you see the same thing as me on the screen?
Yes.
Huh. Can you scroll down? This has been executed. Oh, okay. I'm going to ask you a favor. So do you mind to go to edit, view? Yes. So can you go to edit and come down here and say clear outputs of all cells? Can you click on that? that's much better. I mean, you can, if you don't want to wait for me, you can go the document down and keep executing things. Can you guess what it's going to do? If I execute this, it's going to say hello world, right? That's probably pretty easy to understand. It's the easiest code we can do, right? So if you execute it, now just below it, it printed hello world. So one thing you need to notice, right? When you execute code, the cell, we have a cell of code, and then we have a display just below it. When we did that document and we asked, can you give me a plot of the temperature, it created a display just below it just to show you the graph. So even with text, if you try to print something on it, that's what it's going to do. It's going to create a display just below it. So you can do math with R, just with a calculator. So whoever did the homework that you're supposed to do, you already know this.
Shift enter.
Yep, you can shift enter. You can modify the cells. Feel free to modify them, play with it. So this is a document you can play with it. Right, you can do really complex formulas. And you get the answer of the universe, right? Does everybody knows what a variable is in programming? If you did the homework, you know. But I don't know if everybody does.
We can solve now. We are given that will give you answer now. Suppose we can add our own. Suppose like this is the formula that is solving itself 3 into 3 plus 4. So we are getting answers like 30. So
Yeah.
if I want to do my own formula,
Yeah.
I can add the
You
cells.
play with it. Yes. Yeah. You can add as many cells as you want. So if you want to create a different, I don't know. Yeah. I can see who didn't do the homework here. Oops. I'm assuming you know what is happening here, right? So if we assign a variable to a letter like x equal 5 or x arrow 5, so the traditional R uses this way to assign values to variables. And if I want to print that x, what do you think is going to happen? It's going to print 5. It does the replacement.
Maybe I think it's so x will be 5.
Yep. So you can use x from now on for whatever you want. X is going to be equal 5.
So here we can give command. So we define our x, and then we can go downwards.
Yep. Yep. So the x has been defined by the whole document now. We are reviewing the homework, which we're supposed to do that, right? Because we already done the homework. You already know all this. So every time you see the traffic light, I'm asking you to create something, to code something. So you're going to have to type. If you go too fast with the shift enter, you're going to be missing the answers because the answers are quite obvious in the cell previous to this one. Yeah, everything is installed, yes.
This is a traffic light signal, right?
Yeah.
I'm trying to do this exercise. I'm bold about bold, right?
Oh, you don't, skip that part if you want. We already done this. Go to the, yeah. So when you are coding or creating vectors or creating data frames, it's always useful to print them or to show them on the screen to be sure that you created the right thing. So, and here we are creating a plot, but if you want to be sure that the data frame that we named DF is being created properly, you can always print it on the screen and be sure that it is correct, right? That is always useful. Visualize, visualize, visualize. Everything we do in bioinformatics, you need to visualize it probably, or any kind of job you do. It's always good to visualize the data. Yeah, okay. It wasn't complicated, right? So the exercise, the first one is really simple. I only ask him this, right? To create a plot and start with dots, with lines. Sorry, what?
I didn't get it. How to create a line diagram?
How to create,
Yeah.
sorry, again. Let's start this document for now. So 2 things we need to do. If you have the chance, please do the homework about R. It's going to be really useful for you for tomorrow to follow the whole document. So please do that. If we have time, we can go back to this. If you're curious about the rest of this document, we can do it in another time. So you actually can leave it open. It doesn't matter. Or you can close the document. It automatically, it's been saved for you. So if you open it later, it's going to be in the same state. So one thing that I want to notice about documents is that you see that green dot next to it? It means it's still running. It's active. So if you have code running, you can close the document, the code will be still running for you. So actually shut it down, you need to be explicit. So you could do right click and down here at some point, shut down the kernel for that document, and it would shut down whatever code you're running in it. I mean, you don't have any long code running there. So when you execute a cell, that was done. There's nothing long. But imagine you have something that runs for hours. You can keep it open and close the document, and it will still run in the background. What I want you to do is to open this folder called FastQC. There's a new Jupyter notebook in here called FastQC something else. So if you double click, you're going to see it. I'm going to go relatively fast through it. So instead of you reading it, I'm going to try to explain the document myself. So this document is a bit special. What is the thing you notice? What is the kernel? What is the kernel in this document? Python. This is not R anymore. Why I'm using Python? Because I'm using a trick. So all the code in this document, you could type it in a terminal and it should work. So if you feel comfortable working in the terminal, you can just copy and paste or type the commands in here and work in the terminal. So the trick here, this is not Python code, it's just command line code, it's bash. So the way to do it is, on top of the cell, you're going to see constantly this. This is a way to tell to the Jupyter notebook, this is not Python anymore, this is bash code, whatever is underneath. It needs to be on the top. So you're going to see it constantly in this document, this bash, what they call it magic lines, or the magic cell, you're going to see it on the top. Another thing, there's 2 things that I notice people get confused when we work with this. The first one is, this is code, right? We know this is code because it has the brackets, and if I go to the cell, I know it's code. If you see the brackets immediately, you know that you're dealing with code. Although you think, oh, is this a title because there's a symbol of a number here on top? No. In coding, in R, and other language, it's quite common that if you want to add comments to the code, you use that symbol in front. So when you execute it, this is not executed. You're telling to the kernel, this is just text that I put in the middle of the code. Another thing that I see people confused. So in bash, the way to assign variables is with the equal symbol. Right? Similar to R, we have the arrow. The equal also works in R. So the variable name cannot have white spaces. So our main enemy in this workshop is white space. Never use it for anything. For file names, folders, variable names. Forget about white spaces. They don't exist anymore for you. Try not to use them. So generally we use short names, but you can be really explicit with a variable name. You can say, in this variable, I'm storing something. That could be the variable name if you want to. It's very long, but you do it. This is just to show you that we are executing bash code. You don't have to learn this bash code, but if you execute it, you're going to get this thing. Echo in bash is the same thing as print in R. So basically what we did is assign hello world to this variable and then print it. Now, when we execute command line code, right? So if you go to whoever used the terminal before or another time, you execute a program, just type in the name of the program, right? You open a terminal, navigate to the directory where your program is, type the name of the program, and sometimes you give a bunch of parameters, right? And the program executes. That's how normally we do it. So here we have 2 types of programs. One called ls that prints whatever is in your folder. There is another one with extra parameters, right? We're going to talk about 2 things here. First, do you notice a pattern about how we do, and you know why we sometimes use 1 dash or 2 dashes for parameters?
See what is inside that folder, the content inside the folder.
Well, ls to know the content of the folder, but why I'm giving a parameter with a single dash l, but here I'm using 2 dashes instead of 1.
One heading, two heading. They are the, we have been getting pipelines, the hierarchy of the data.
No, no. Let me rephrase it. I'm not asking about ls. I'm asking why I'm giving. If you pay attention, sorry, I'm going really fast, but I want you to learn this thing. So, in here happens the same thing. Sometimes I'm using a dash. Sometimes I'm using 2 dashes. My question is why I'm using 1 or 2.
So that represent the hierarchy. So DTA is coming from T, right?
No, it is not a standard. There's no rules. So there's no hard rules about it. But generally, when you want to give a parameter with a single letter, you use 1 dash. When you want to use the full name of that parameter, you use 2 dashes. So this is quite common. So if you are running, you will see later that sometimes we use a single dash, sometimes we use 2 dashes. So we can keep executing. Another thing, we're going to go through these tomorrow again, probably at some point, and I'll try to go a little bit slower, right? So we show you a few parameters. ls has a few of them, right? How do I know about them? So there's no hard rules about it, but one rule is you can just type the name of the program, and sometimes the name of the program tells you, oh, these are my options. What are the options that I have? What are the parameters? That could be one thing. The other is quite common that you can add the parameter -h or help if you need to know more about the program. That's quite common in the command line. In command line also for the system calls, they have a manual, and then you can call man and the name of the program, and it gives you a manual. All of this is impractical at the end of the day. So the practical thing is to just Google. Say, okay, Google, can you tell me what these options are for these parameters, and you get the answer. And nowadays, Google incorporates AI, right, or the machine learning, the LM, and so you have them there, and it gives you an answer, and why you use these parameters and why not. So feel free to open, if you have doubts, just use Google to know what options we have for each program. Oh, sorry. The other thing you probably notice is every time this is the document for ls, right? It's not practical, right, to read all this. It's easy just to go to a Google and ask what it means -- color. Another thing I want you to learn quickly is all these cells, when they're really long, you can scroll inside of the cell up and down. But if it gets annoying, you can extend it so you don't have to scroll, or you can even hide it completely so it doesn't bother you. Okay, to simplify it, I'm going to skip, and we'll go back this later. So now what we're going to do, 2 things. How we organize all the data we're going to see about samples is going to be organized in the same way. So generally, we're going to have a sample folder, and within that sample folder, we're going to have an align folder, and then we're going to have the sequences. Don't worry about what it means. We're going to learn. We'll try to find time to explain that format, but this is just how we store the sequences. Okay? Yep. This is a file with the read one, as Bonnie was explaining. When you sequence a fragment, you can sequence with a single read or pair end. You can have read one and read two. We generally store them in separate files. You could even store them in a single file if you want to, but this is quite common that you have read one and read two in separate files. So this is the type of structure you're going to see through the course. Every time we talk about samples, you're going to have them inside of folders. Now what we have here, I didn't name them with sample names specifically, but these are samples. So the folder that says RNA-seq is actually a sample. So if you double click on it, you can do it. You're going to see what I said. You have an align file folder, and if you double click on it, you have, sorry, it's hard to read because I'm going to have to extend this. You have the read one and read two for that sample. Yep. So vertical line here, this command, can anybody guess what it does? We learned this a moment ago, head, right? We use it in R, where it prints the top 10 or top 6 lines. So this is also a pipe. You remember the symbol with the percentage, bigger than percentage, that's a pipe in R. In bash, this is the pipe. So zcat, what it's doing here is I'm compressing this and giving the output to head. So if we do this, what do you think is going to happen? Yeah, uncompressed, well, only on the fly. It's not actually create an uncompressed file, but it's going to print the top 10 lines of the file. Again, we're going to go through this another time when we have time about talking about the format because we are really short on time. So I'm going really fast through this. But this is how the FASTQ file looks like if you open it. We don't open the files because they're way too big. They are gigabytes of size big. So don't try to open a file in a Word document or anything like that because it's not going to work. They're humongous. But you can do exercises like this that show me the first 10 lines. So I want to look that the file is correct or it looks like it's well formed. So just to go fast with it, the only thing you need to know about for now in this file is every read, where Bonnie was talking about a read, one sequence, is stored in 4 lines within this file. It's a file name here on top, the sequence itself of the read, the 3rd line, don't worry about it, it's just part of the format. The 4th line is this thing here that looks like a read to. It's not actually a read. It's a way the quality scores. It's the way we have to store quality scores. Don't worry about it, but do you remember Bonnie was mentioning the Fred score or the Q scores and all that? That's the way that it gets stored. So the idea of the exercise we're going to do now was to, and you can do it if you want to, is to evaluate the quality of the sequences. Right? To evaluate the quality of the sequences, we use a program called, quite commonly, it's quite common, this program that we use constantly. It's called FastQC. So what we're doing now is telling FastQC, we're giving the FastQ sequence, right, the file, and it's going to give them a report of the quality of the sequences. By the way, you can follow, we're following the document too, you can execute the lines. So we're going to get some output on the screen telling us, oh, I'm processing the reads. So now I finished processing. And what we have, if you go to the folder, in the unaligned place, there are more files now. One of them is an HTML. So if you double click, Jupyter Notebooks can read, or Jupyter Lab can read HTML files. So this is the output of FastQC. What you see on the screen now is the output. And what we can do is go through one of them. And then we'll figure out a way to, we're going to explore different FastQCs between groups. We can discuss them. How much I have? 20 minutes? Well, before 5, we need to finish, right?
Yeah, so always actually, once you get your sequencing data, the first thing that you need to run is to run a FastQC and make sense out of, don't rush into just running the analysis. So just like my PhD supervisor used to tell that one of the worst things is to, like, if you got divorced, the worst thing to happen, divorce is not the worst thing. The worst thing is next day morning the first person whom you see going and marrying to that person is the worst thing. So similarly, the issue is don't rush into your sequence, rather than first look into the sequence, understand that how good the data is, make sense out of the quality, and then rush into sequence. And this is FastQC will give you a handle on that.
Yep. He said half-truth. Right? We sometimes skip that step. We see that something is wrong, and then we go back to look at it.
That is more like a practical thing.
Yeah. So if you run the FastQC, if you try it, you will see you get an HTML file. When you open it, you get a report out of it. So the report is going to divide it in different sections, and you're going to get a green check if everything looks good. You may get an exclamation mark because there's something that is not that good, or you may get some sort of red mark telling you there's something wrong in there, right? One of the things, the message we're going to try to get through this half hour that we have left, or less than that, is that the report is not always right, in the sense that we have RNA-seq, we have methyl-seq, we have whole genome sequencing, we have a lot of types of sequencing. This FastQC is thought about sequencing for whole genome sequencing, basically. But depending on the type of sequencing you do, the report may look like slightly different. It doesn't mean the data is wrong. It's because the report doesn't know exactly what you did, right? It's reading sequences. And we'll see what the differences are depending on the type of sequencing you do. Should I use the mic, maybe? It's on? Oh, it's not on. Okay, sorry. You get some basic statistics. Basically, here it's telling you, I have 100,000 reads, sequences on that file. It tells you the length of the read, the GC contact, nothing special, right? There's nothing telling much about how the quality it is. Probably the most interesting plot that you're going to see is the first one. I'm going to have to, which is the quality per base. So on the x-axis, we have the length of the read. And in the y-axis, we have the quality. Illumina goes from 0 to 40. 0 mean really bad quality, 40 mean the highest quality, and represents the how confident am I that base is correct or wrong. Actually, yeah, it represents the percentage of being incorrect. So what has been done here is accumulate every read. So you have a box plot for every position 1, right? So we have a bigger one here. So the box plot, this represents, I think, the 90% of the reads, or the base 75 falls within high quality. There's a 10% below this that is low quality. So you have a box plot for every position in your read, in your all reads, right? And 100,000 reads that you have. So everything on the green area is supposed to be good. So the first thing you need to notice, what quite common, at least in older sequences, is the quality drops at the end a little bit, right? So one thing you could do if you want to improve alignment or maybe another process is trimming the low quality bases at the end, so you have a shorter read but it's higher quality. This, we barely make use of it, but this represents the flow cell. Generally, we don't look at it that much, but when you have, it would tell you if some bad quality reads are together in the flow cell, it would illuminate red, saying, oh, it seems that your flow cell, there's an area that is wrong with it, because all the reads that are bad come from the same area. So it's supposed to be random, right? But it's not sometimes. Another way to look at the quality scores is to show an accumulative curve, right? Where it tells you, oh, most of the reads are just high quality. That seems that everything is good. So this, for example, here. This is the GC content, again, per base. The average of the GC content per base. So if we know that it's human, so basically, ANT and CSG is supposed to be complementary. We know the composition of the human genome. This plot should represent the human genome. So the first thing you notice, so we have a question mark, exclamation mark. It's telling you there's something wrong here. And it means this thing here, right? At the beginning of the read, it seems that it's not fully random, right? It's supposed to represent the human genome. We can say that more or less every base should be represented 25% of the time, right? But why it is not at the beginning of the read? And it's telling you, oh, there's something wrong here. That's what it tells you FastQC. But you know, this is our RNA-seq sequences. So this is normal for RNA-seq. So they are not completely fragmented. When you work with RNA-seq, that's not totally random. So the first, generally between 6 to 10 bases are not random. So they are not represented the genome. They represent the transcript. Whatever fragments come from the taxon, that actually represent the N6. I'm not going to get into details, but this is common on RNA-seq. So what I'm trying to tell you here, if this was a random sequencing of the genome, there would be a flat line from beginning to end. Another way to look at the GC content, it's nothing much to see here. The N content, right? You're supposed to have 0 N's in your, N means represents any base. So in theory, there only should be A, C, T, and Gs. There shouldn't be any Ns. So if you have a lot of errors in your sequencing, you're going to see a spike of Ns or something like that. So this looks good. All our reads are the same length, so nothing to show here. This is another interesting one. The sequence duplication levels. So in RNA-seq, or if you do whole genome sequencing, the sequencing duplication should be zero, almost zero, because so all your reads should be one time. You shouldn't have reads being duplicated. So if you have, sorry, this plot, what is represented is how many times you see a read, right? Or the percentage of times you see one copy of one read, most of them should be in the 1 or 2 copies, meaning no, you don't see duplication. If you see a spike later on, it means there's a read that is being duplicated a lot of times. There's something wrong with your sequencing. In theory, in whole genome sequencing, you should have every read only represented once, covering the whole genome, or maybe twice or 3 times. In RNA-seq, it's not the case. So sometimes you're going to see spikes of duplication because it's expected that a transcript being highly expressed is going to be sequenced a lot of times. You should see peaks on it. Probably we don't have a red mark here just because we only have 100,000 reads. But if you would have the whole file, you're probably going to see a spike at some point.
Do we need to correct these things? Or, when there are a lot of duplicates?
If it's RNA-seq, no, right? Because it's expected. If it's genome sequencing, you probably cannot do much except removing duplicates later on, right? So there's a process of removal of duplicates. You can do that.
So one of the reasons why duplicates arise is because of low input quantity. One of the main mechanisms for how it happens. So that means you have fewer number of molecular diversity and same molecule sequenced again and again, and that is amplified through PCR in the library. And the only way you can do that, as Francis mentioned, you remove the duplicate reads and only retain one read from all those. But again, if you have a very high percentage of duplicate reads, it becomes tricky, because the idea is like, suppose you have 20% duplicate reads, then you can do some extra sequencing and you can cover that 20%. But if you have 50% read, then now you have to make a decision that whether you want to do that extra sequencing for 50% or you want to say, I want to redo the plot, based on how this is the data. I will
Yep.
finish this and
Sure. Yes, sequence duplication is green in this case. I'm surprised because it's RNA-seq. We should have some duplication. I'm feeling it's because we don't have the full file. That's the reason we don't see the duplication yet. You would have some overrepresented sequence here in the bottom. In some cases, it's normal. It's quite common you're going to see the adapter sequence being duplicated. That means we sequence the adapter, Illumina adapters that is part of the library preparation. You sometimes see them. Okay, it's not a problem. You can throw them away later if necessary. You can trim them off. Here we're going to see something slightly different, right? So this is the sequencing read. And if you see adapter sequence showing up, it tells you on what base you start seeing that adapter sequence, right? So if you have really short fragments, you're going to see a line going up, because meaning we sequence our fragment and then we went over the fragment and we start sequencing the adapter. And here it tells us that, yeah, we have some sequencing adapter at the end. It's not what represents maybe 5% the most. It's not that a problem. You can still trim it off. So this is the type of report you're going to get. So we have a few minutes. What I want you to do is, so if you go to, sorry, day 1, FastQC, you have some examples here. Oh, sorry. Yeah, you see them. You have, for example, we can look at, should we use methyl-sig, for example, right? So here we have another example. Now we see red marks, right? In some cases. Quality, it seems okay. It doesn't look like good as the previous one, but it marked as green. Okay, quality is good. Nothing to see here, nothing to see here. Now, okay, it seems we have a problem in in the GC content. Is it a problem? We are doing methylation sequencing. Is it okay? Is it bad sequencing or is it good? What do you think? FastQC is telling you it's bad. Is it?
Yeah, anybody know how we do methylation sequencing?
It's not wrong. So you do a conversion of G to C's, right? So it's expected, actually the other way around, right? Well, depending on whether you're doing C to T, yes, sorry. So you're going to change the frequencies. So this is okay for methylation sequencing. Even the FastQC is telling you there's something wrong with it, right? So what I'm trying to tell here is, yes, the context is very important. Whatever type of sequencing you do, you need to understand that FastQC may give you an error, but it's not an actual error. I don't know, it's giving us 2 more errors. We can look at it. So what we said before, right? It seems we have some short fragments, right? Fragments that ends at 40, and then all of a sudden we get some Illumina adapter. Okay, that may be a problem, yes. So what you can do is try to trim those reads that have adapter and make it shorter. That could be an option. So what other we have here? So microRNAs. Okay, quality looks good. Per base sequence content looks terrible. It may be normal though. So microRNAs are going to be really short, only 20 bases. Some of them they're pretty conservative. They have sequences that may be constant. So maybe that is normal. Sequence duplication is going to look terrible, but it's fine too because again, they are so short that when you do sequences so long, you sequence the RNA plus the adapters. So that's normal. So again, the important thing here is, as we said, you need to know the context.
Okay, so Francis, we can actually stop for one. I have one other thing. Suggestions for you guys. It depends upon you. So we have a talk from 5 to 6, and if you want to stay back for extra 1 hour and do some more exercise on fast QC, I think that should be fine. What do you think?
So it could be
but it depends upon you.
6 to 7. Yeah. So people who want to stay back from 6 to 7, they can. We will be here around. But we have an online lecture from 5 to 6.
So it depends upon how you want to.
We'll review some of those things. FastQC, we're going to be running it at least 2 more times in the next days. So you will learn how to run it. I mean, it's not complicated. So we'll do. And we can review a little bit more of those things tomorrow if we have time.
Even if you want to practice more, we are happy to stay back. We have to spend more time. We have no problem.
I can stay back. Most people agree.
Yeah, it's up to you because many of you have to probably travel back to your homes and you have other commitments. So we know that. So you are answering your question. What happens?
So one more thing, by the way, if you have questions and you don't want to ask them now, you can send us an email. So our plan is every morning, the first minutes in the morning, we review the previous day. So you can send us questions now or ask them tomorrow morning, the first 15 minutes. So we try to review whatever we did the previous day. So this is the FASTQ format. I was showing that every read is 4 lines, right? The first one okay, here we go. The first one is the ID of the read. They always start with an @ symbol here. These numbers actually mean something. And FastQC makes use of this. So this thing, for example, this thing here is the flow cell number, or the ID for the flow cell, the thing that we put in the machine with the samples, that's the flow cell ID. And this represents the location of that read in your flow cell. Remember, we show FastQC makes a plot, and it tells you if there is a lot of reads clustering in one section of the flow cell, lights up. So thanks to the location, it's able to do that. So actually, this has a meaning. If it looks like random, for example, this means it's a read one. This is some the barcodes that we use for samples. Indexes for the samples are encoded here.
So this is a dual index sample, so that is why you have 2 indexes. So dual indexing means you will have index on both sides, and so that actually saves you both and separated by that first sample.
So it's used to separate samples.
Yeah.
So this is the barcode for every sample. The second line is the now just the basis, right? The sequencing itself. The third line, again, always starts with a plus. So the interesting line probably is the fourth one, and it's the way that the quality scores are encoded. Probably you heard about the ASCII codes. That's how the keyboard or the letters in your keyboard are encoded. They use numbers. So actually, this letter represents a number. It actually represents a number from 0 to 40. They apply a correction of 33. So actually, F must be 70 in the ASCII code. If you subtract 33 to 70, you get 37. So F represents a quality score of 37. So that's the way they have to encode it. So it's simple because every base has a single code or a single letter to assign a number of 37, for example. In this case, there's a lot of Fs. So the colon means another different code. So you look at the ASCII code, you would manually calculate if you want, and you know what quality score represents. You don't have to. Again, this is an exercise that we're doing just to understand how the FASTQ format. The only thing you do is to use FastQC, and FastQC does the job for you, right? But the quality scores are really important, so we need to store them together with the sequences. It's not enough with the sequence. He mentioned that the errors are random. That's not also completely true. In Illumina sequences, they're not totally random. And that's also a problem, right? But if you use PacBio and other sequences, they're supposed to be completely random. That's it?
Yeah, we'll connect the Zoom now, so, and then we'll come back. We can come back. Vaishali, right? Vaishali, no? Yeah.
You need this?
He connected. I don't know which one.
Oh, you can use this. This is mine. You can use it.
the next lecture will be online. This will be delivered by Dr. Gyaneshwar Chaube. He is a professor at the Department of Zoology at Banaras Hindu University. So Professor Chaube had received his training as a project assistant at CCMV Hyderabad and also held as a position as a PhD student and scientist and senior scientist at the University of Tartu, Estonia.